Okay, so today we're going to start with the collagen. And first thing you should know that it is the most abundant protein inside the human body. And collagen actually provides a tensile strength to our body tissues. Okay. Uh, you know, Once you get the wound, after your wound, become the scar tissue. The scar is actually a collagen. Okay. That provides the strength, the remaining strength to that tissue. Okay. So collagen is very necessary for every extracellular matrix inside our body. It provides some the strength, the tensile strength that is required. Okay. For example, after the myocardial infarction, the scar tissue you will left with inside your heart is actually the collagen. Okay. So based on the types, we have type 1, type 2, type 3, type 4 collagen. That type 1 collagen is the strongest actually. It provides a real strength to our tissues. The bone, for example, is made by the osteoblast. It is a type 1 collagen. Skin, it provides a strength. It protects. So skin is made up of type 1 collagen. The tendons are made up of type 1 collagen. The dentin that is found in our teeth is made up of type 1 collagen. The fascia, the cornea, the late wound repair, scar tissue is actually known as the late wound repair, or the scar tissue is made up of type 1 collagen. Sometimes, Stefan gonna ask you about this scenario where they will create like a scenario of MI, and they will ask you after 14 days, you're left with something as scar tissue, and they will ask you what other type of tissues are made up of type 1 collagen. Okay, and they will ask you this. So they will give you some options like cartilage, the reticulin, the bone, the skin, and you will choose them. Okay, I hope you guys understand my question. They will describe your scenario of myocardial infarction, and they will tell you after 14 days what type of tissue can be, can represent this tissue. This is scar tissue. They will
Can't
give you
hear the
multiple
speaker's
options.
voice. So,
Can you hear it now?
can anybody speak to me back so that I can understand that yes, my voice is being heard?
Yeah, I can hear you. Can you hear me? Am I audible to everyone?
Yes, sir.
Yes.
Yes.
Okay. So am I clear how questions could be presented in this type? Yeah. And you should also know that the osteogenesis imperfecta type 1 is actually a disorder of type 1 collagen. And in this type of disorder, there is a decrease a production of the type 1 collagen that leads to osteogenesis imperfecta. We will study that later in the next page. You should know what type of disorder correspond to what the type of collagen. Okay. Then comes a type 2 collagen, which is found in your cartilage, including your hyaline cartilage, the vitreous body inside your eyes, and the nucleus pulposus, which is found in your intervertebral discs. Okay. These three are made up of type 2 collagen. Then comes your type 3 collagen, which is found in your reticulum, which are the reticular fibers in your skin, blood vessels, uterus, fetal tissues, and the early wound repair. Remember the granulation tissue after the wound repair? It is made up of type 3 collagen. Type 3 collagen is kind of a stretchy, everything you can imagine which is stretchy, like your blood vessels, like the uterus, like the skin. Your skin is so much stretchy. So everything that is stretchy are made up of type 3 collagen. Sometimes Stefan gonna give you an option, not like the skin, blood vessels, uterus, but they will tell you that reticulin or the reticular fibers. So do not get confused if they give you this reticular fibers in the option. This corresponds to type 3 collagen. Okay, there is a disorder or the syndrome known as the Anhedonia syndrome. It is also a disorder of type 3 collagen. We will study that later also. And then finally comes a type 4 collagen, which is present in your basement membranes. Basement membranes, I hope you all know, it is also known as a basal lamina and the lens. These two things are made up of type 4 collagen. And you should know that defects in the type 4 collagen. First is the X-linked dominant syndrome, which is the Alport syndrome. You will study that in the renal chapter. This Alport syndrome is a type 4 collagen defect. And then there is this autoimmune disorder known as a Goodpasture syndrome. We will study that later in the renal chapter also. This is also a type 4 collagen defect. Okay. Then let's talk about the production of the collagen, how it is synthesized in our body inside the cells. So what cells actually synthesize the collagen? These are your chondroblasts or the osteoblasts, okay, in your cartilage or the bone. They actually synthesize your collagen, or the fibroblasts. Okay, these are three type of cells that synthesize the collagen. So first of all, you should know what amino acids are present in the collagen. And these are the glycine, proline, and lysine. You should also know that glycine content is always fixed, and it is 1/3 of the total content. Doesn't matter how much proline is there, lysine is there, glycine is always going to be 1/3. So we always don't it like glycine X and Y. X and Y could be a proline or could be a lysine. So whenever a step one question asks you what is the most abundant amino acid inside a collagen, you will always answer the glycine, because the content of lysine or proline is not fixed. It is variable, but the content of glycine is fixed, which is 1/3. So it is the most abundant amino acid inside the collagen. And they can also ask you why it is so, because the glycine is so small that it can easily fit inside the in between the lysine and the proline, and that's why the glycine is very abundant in it. Hydroxyproline, which is formed after the step of this hydroxylation, is used for the lab quantification of the collagen. Whenever you need to know how much collagen is present inside your body, you will use the hydroxyproline for that. Okay. Then comes your step synthesis. First of all, you have this backbone of glycine, lysine, and proline. Then comes a step of hydroxylation, where the enzyme hydroxylase, either the lysyl hydroxylase or the proline hydroxylase, puts up these hydrogen groups over this backbone, and you will left with this hydroxylysine and hydroxychlorine. You should also know this enzyme requires a vitamin C for this step, and so if there's a deficiency of vitamin C occurs, it is known as a scurvy, and it will left you with multiple symptoms. We will study that later in the vitamin chapter also, that bleeding, gums bleeding, and a poor wound repair, and all these are symptoms of your scurvy. And why is this so? Because the collagen requires the vitamin C for its production. Then comes a step of glycosylation. Glycosylation is basically, I told you, the post-translational modification. Glycosylation is basically the glucose residues are on the protein. In this scenario, actually the lysine has this glycosylation. There is a put up of a glucose on the lysine, and you will left with the hydroxychlorine and glycosylated lysines. So the three chains, these are the alpha chain, one alpha chain. Similarly, another alpha chain and another alpha chain came closer, and they form a triple helix, which are joined together by the hydrogen or the disulfide bonds. You should know about this bond. The hydrogen bond and the disulfide bond are the two bonds in the alpha triple helix. And this is the thing where which leads to the osteogenesis imperfecta. If there is a defect in the formation of this triple helix, this will lead to the osteogenesis imperfecta. Step one gonna ask you this, so you should remember this guy. Okay, a problem in triple helix leads to the osteogenesis imperfecta. Glycosylation occurs only at the lysine, but hydroxylation occurs both in the lysine and the proline. And finally, the glycosylated lysine and the hydroxylated proline will exit out of the cell. The process is known as the actocytosis. They will enter the extracellular space. Here, the collagen is known as a pro-collagen, not the collagen. Likewise, I told you there is a post-translation modification and the protein needs to be activated, right? So here, the collagen is inactivated, known as a pro-collagen. Now, after the proteolytic processing, their N and C terminals are cleaved off. There are disulfide-rich ends. They are cleaved off, and now the collagen is known as a tropocollagen, which is insoluble. And finally, the cross-linking of this tropocollagen occurs, and the enzyme here is your lysyl oxidase enzyme, which is a copper-dependent enzyme. It will take up a collagen, makes a cross-linking with them, and finally causing your wound repair. Then there is a disease known as a Menkes disease, in which there is a deficiency of copper. And as I already told you that the lysyl oxidase is dependent upon the copper for its functions, or a deficiency of copper will lead to the deficiency of functions of this lysyl oxidase. So you will get the poor wound repair, and a disease known as the Menkes disease. You should also know about this Ehlers-Danlos syndrome. It is of two types. One defect is in the intracellular defect, and one defect is the extracellular defect. The intracellular defect is basically a defect in the lysyl hydroxylase enzyme, where the hydroxylation is not occurring in the lysine. This leads to the Ehlers-Danlos syndrome. Similarly, the extracellular defect is basically the proteolytic cleaving process. If the proteolytic cleaving does not occur, then the collagen will remain the protocollagen. the pro-collagen, it will never get activated, never take part in the wound repair or anything, and you will left with the Ehlers-Danlos syndrome. Okay, you should know these two things regarding the Ehlers-Danlos syndrome. It is of two types. It can be due to two things, whether the intercellular defect or the extracellular defect. Stefan gonna ask you this also. Okay, any doubt in this page, or should I move ahead? Should I move forward now?
Yes.
Okay, no doubts. Okay. Oh yeah, where does the collagen synthesis occur, guys? Inside a cell. Where does the collagen
Rough,
synthesis occur?
Rough
Huh?
rough endoplasmic
reticulum.
reticulum.
Yeah. Like I told you, every secretory protein is synthesized in the rough endoplasmic reticulum. Now comes a disease, the osteogenesis imperfecta, which is a disorder of your bone. You should know, guys, that this is not a defective collagen production, but a decreased collagen production. The collagen will remain absolutely normal, but it has a decreased production. Like I told you, if there's a promoter sequence defect, in the previous pages, I told you about this promoter sequence defect. If there's a defect occur or a mutation occur in the promoter sequence, then you will get a decrease production of mRNA, right? And it will lead to the decreased production of proteins. This is the same thing. Osteogenesis imperfecta is actually a promoter sequence defect, and that's why it leads to a decreased production of a normal type 1 collagen. You should know two type of genes are involved in it, and it is a COL1A1 gene and a COL1A2 gene. And anyone can tell me if two genes, different or same mutations are happening at two places in these genes, what this is phenomena known as? These are producing the same phenotypic effects, the OI. What does this phenomena called? If you're getting multiple mutations at multiple genes, but same phenotypic effect, locus heterogeneity. You will study that later in the genetics part. Locus heterogeneity. So osteogenesis imperfecta and the Ehlers-Danlos syndrome are the very classical example for the locus heterogeneity. You should remember this fact. So what are the manifestations of this osteogenesis imperfecta? Because your bones are very weak and brittle, they are easy to fracture. So multiple fractures are the first history of the patient. There's a movie on it also. I forget the name, but yeah, there's a movie. I will send you the link if you want to watch. So multiple fractures are the first history. There's a bone deformities, and bone get fracture with minimal trauma. So sometimes a doctor might think it a case of violence, child abuse. So you should remember this fact. Then you have this blue sclerae. Why this blue sclerae? Because there are some veins behind our choroids, known as the choroidal veins. And I already told you that type 1 collagen is found in your cornea, as you can see here. So if the deficiency of collagen occurs in your cornea, the veins will become very visible, and it will show you as the bluish appearance here of the sclera. As you can see the eye, they will show you the picture and they can ask you the question over it. Should remember this picture very carefully. And likewise, I told you the uh, um, collagen is also found in the teeth, the dentin. So you will also get the tooth abnormalities in the osteogenesis imperfecta. Lack of dentin is known as the dentiogenesis imperfecta also. Your tooth will wear off very easily. And then there are also bones inside your ears, the malleus incus and the stapes. They will also get affected, so you will get a conductive hearing loss. Okay, so there's two type of defect. First is the decrease production of the type 1 collagen, and the second is the decrease production of the type 1 helix, type 1 collagen triple helix. You should remember these two facts about the osteogenesis imperfecta. So what are the symptoms? The bite, multiple fractures, blue sclerae, dentinogenesis imperfecta, and the hearing loss. It can mimic the child abuse, so be careful about it. Okay, then the Ehlers-Danlos syndrome, which is basically a defect in the type 3 collagen and also a type 5 collagen. Likewise, I told you the type 3 collagen is very stretchy. Okay, and if you get a defect in this stretchiness of this type 3 collagen, you will get symptoms regarding the skin, the uterus, the blood vessels. So these patients have very a tendency to bleed. They get very easy bruising. They will get a very hyperextensible skin, a hypermobile joints. Okay, it kind of multiple types: the classical type, the vascular type, the hypermobility type. The hypermobility type is the most common type, which is you will get the symptom of very hypermobility of the joints. One thing that you should remember is my voice is echoing actually.
Someone's mic is.
Yeah, someone's mic is on. It's echoing. Okay, now? Yeah. Okay, so where were we? Yeah, you should remember the thing about this anhedonia syndrome, that it is also associated with the Barry and the aortic aneurysm. Okay, please remember this fact: the Barry and the aortic aneurysms. Later in this chapters, we will study about them, and we will study about other diseases which are associated with these two aneurysms. So you should know that one of them is the anhedonia syndrome. Okay, it could be an autosomal dominant or an autosomal recessive disorder. The most common presenting complaint is a joint dislocation or the hypermobility of the joints. You should know, because their skin is so much stretchy, so much fragile. So the suturing of the skin becomes so much difficult. Okay, you should know about this fact also. Suturing of the skin becomes so much difficult in them. And then the hypermobility type is the most common type. Like I told you, then there is a classical type which is associated with the defect in the gene COL5A1 and the COL5A2. It is a type 5 collagen defect. You will get the joint, which is a hypermobility of the joint, and the skin symptoms, very stretchability of the skin. As you can see here, the skin become very stretchy and the joint become very hypermobile. These two things are associated with the classical type of Ehlers-Danlos syndrome. Only the hypermobility is associated with the hypermobility type. Then comes the vascular type. Like I told you, the blood vessels has this type 3 collagen. So vascular type of Ehlers-Danlos syndrome is actually a type 3 collagen defect with a defect in the gene COL3A1. So as you know, this type of collagen is found inside your skin, blood vessels, uterus. So what you will get a gravid uterus, a very easy rupture in the pregnancy. Okay, you will get multiple bruisings, easy bleeding, and your skin become very fragile, very stretchy. Okay, it will affect multiple organs and the muscles also. So this is your vascular type of uh, Ehlers-Danlos syndrome. Okay, like I told you, there is two type of defect: the intracellular defect and the extracellular defect. Intracellular defect is due to the lysyl hydroxylase deficiency, and extracellular defect is due to the uh, deficiency of NC cleavage. Okay. Any doubt in these two disorders? Should I move forward? Anyone? Am I audible to everyone?
Yes, sir.
Yeah. So should I move forward now?
Yes, yes.
Okay. So next come the Menkes disease. Like I told you, the lysyl oxidase enzyme dependent upon the copper for its function, and it is required for the cross-linking of the collagen. So if there's a deficiency of copper occurs, then this lysyl hydroxylase cannot work properly, right? And there won't be any cross-linking of the collagen occurs. So this will lead to the Menkes disease. It is an X-linked recessive, very important X-linked recessive disease. And due to the defect in the gene codes for ATP7A, remember the ATP7B for the Wilson's disease? The ATP7A is for the Menkes disease. In the ATP7B, the Wilson's disease, you will get very high load of copper inside your body. But in the ATP7A, you will get a very low copper inside your body that lead to the dysfunction of the lysyl oxidase. Very low level will lead to the, this easy bruising, first of all. The second thing is delayed wound healing. Third thing, you will get a very brittle, very weak, very kinky hairs. Kinky hairs are these straightened hairs. You will see these as a straightened hairs. You will get the growth retardation, the hypotonia, and the risk of cerebral aneurysms, because your blood vessels will become very weak, right? So you will have an increased risk of their rupture, aneurysms. This will lead to the stroke also, the cause of death in these patients. Okay, any doubt in the Menkes disease? No. Okay. So let's talk about the aging, and the role of UV light in the aging. So guys, there's two type of UV rays. There are many type of UV rays, but we will discuss here about UVA and UVB. Remember I told you about this nucleotide excision repair, the pyrimidine dimers, and the cancer, the xeroderma pigmentosum. That
Yeah.
is a UVB. This is a UVB rays that causes the skin cancer or the sunburn. And then that's that dimer thing. Okay, it is UVB. UVA cannot penetrate deeper into the it can penetrate deeper into the skin layers, and it will decrease the collagen production inside your body, and it will also increase the activity of metalloproteinases, which destroy your collagen inside the body. So what you will get with the aging is decreased production, increased activity of metalloproteinases, increased cross-linking. Okay, you should remember this fact: increased cross-linking with the aging of collagen. Degradation of type 1 and type 3 collagen occur. Okay, there's a thinning of the epidermis, because epidermis consists of the collagen, and if the collagen is being destroyed, its production is decreased, so you will get a thinning of the epidermis. You can feel it's also inside the in the old age looks, and their skin will become very thin and very stretchy also. Skin become their atropic. Okay, so the three things that you should remember about the aging: first of all, the UVA is responsible for the aging. You will get a decreased production of the collagen. You will get the increased cross-linking of the collagen, and you will get also the increased activity of metalloproteinases. Okay, these three things you should remember about this aging: UVA, the collagen production, metalloproteinases, and the collagen cross-linking. You will get a thinning of the epidermis also, and that also can lead to the wrinkling of the skin, because it is low, collagen is very low, so you can also get the wrinkling. Okay, any doubt in previous page? Anywhere?
No.
Okay, then we'll discuss about the elastin. Elastin and collagen are very close to each other. Both the elastin and the collagen have this proline, glycine, and lysine. But unlike the collagen, elastin do not has hydroxylations. Like in the collagen, you will have this hydroxyproline and hydroxylysine, but it's not the case in the elastin. Elastin is actually a very stretchy tissue, that found inside your skin, the lungs, the arteries, the ligaments, the vocal cords. It is very stretchy tissue. So sometimes step one gonna ask you that what actually contributes to the property of this elastin, the stretchy property of this elastin. So you will answer the cross-linking, which takes place extracellularly. This cross-link actually gives the elastin its stretchy properties. Okay, when the elastin formation occurs, like the collagen, it is also a very inactivated elastin. Now, after the extracellular, when it enters the extracellular space, something known as a scaffolding occurs inside the elastin. Scaffolding is like making a protective layer outside the elastin, and it is made up of fibrillin. Fibrillin is actually act as a scaffold, takes part in the scaffolding of the elastin. You should remember this fact: the topoelastin with the fibrillin scaffolding. Okay, and finally, after the scaffolding, the cross-linking of the elastin occurs that gives its elastic properties. You should also know that elastin can be easily broken down by the elastases, and elastases are inhibited by the alpha-1 antitrypsin. So when you get a deficiency of alpha-1 antitrypsin, you will get a very increased degradation of this elastin, and that can lead to the COPD inside your lungs, because I told you elastin is a stretchy tissue that is found in your lungs. Okay, whenever you stretch something, it always like your skin, it go back to its original shape, and that is due to the elastin. But when you destroy it, your lungs become very open, causing the COPD. Your airways will become very dilated. Okay, and along with the COPD, you will also get this liver failure in this case, because alpha-1 antitrypsin being trapped inside the liver cannot go outside. So it will also causes a hepatotoxicity. So we get two symptoms: the lungs and the liver involvement in the alpha-1 antitrypsin. Changes with the aging, I have already told you: the decrease in the collagen production, decrease in the synthesis of the collagen fibrils, and the first aid is wrong here. It says the cross-linking is normal, but actually the cross-linking increased with the aging. Okay, you guys can correct it. Cross-linking increases with the aging. And then comes a Marfan syndrome. Okay, it is an autosomal dominant syndrome, which is due to the FBN1 gene mutation. You should know that it is not a disorder of elastin, but actually a disorder of fibrillin, which protects the elastin. So defected formation of the fibrillin leads to a damage of the elastin, and that leads to the Marfan syndrome. You should know that is an autosomal dominant disorder. And second thing, chromosome number 15 is very important for Marfan syndrome. So now I told you that elastin is a stretchy tissue that is found inside your lungs, arteries, skin. So symptoms you will get are, regarding these, associated with the defect in these organs, right? First, the very important thing that you will get is this pectus excavatum. Okay, the pectus cacarinatum, this notching you're seeing here is a very characteristic feature of this Marfan syndrome. They will describe you a patient who is very tall, like 6'3" feet, or 6 feet 4 inch, 6 feet 5 inch, and they will tell you their flat is foot. They are flat foot. First thing, they are very tall, above 6 feet. They have very long arms, and long legs. Their body to leg ratio or body to arm ratio is very, low. Okay. And someone say something? No? Okay, so their body to arm ratio is very low. Okay, they have very long arms, long legs, and their finger is very long, long fingers, and with the, tapering ends. Okay, and there is something, you will also get a very hypermobile joints. Okay, and there's something called cystic medial necrosis of the aorta. You guys know that there are 3 layers of aorta. Okay, any artery, the tunica externa, the tunica media, and the tunica interna. So what you will get in the tunica media, you will have these muscles, the collagen, and the elastin. Now, due to this, normally with the aging, you will get this thing, the cystic medial necrosis is a normal aging process. But in case of Marfan syndrome, this step is, very speeding up. Okay, so cystic medial necrosis happened at very early age. Due to this cystic medial necrosis, you will lose your elastin, and that's why you will get these aneurysms of the arteries, especially the aortic root aneurysms, and they become very easy to rupture, and the rupture of the artery or the dissection of the aorta are the cause of death in these patients. You will also get the mitral prolapse in these patients, which is the second most common cause of death, the cardiac failure. And something was a subluxation of the lenses you will see in the Marfan syndrome. What is a subluxation of the lenses? It is basically a dislocation of the lens. As you can see in the image, if it is a normal eye, then this red area covers, which is a lens. This red area covers everything. But in case of subluxation, your lens will go outward and upward in case of Marfan syndrome. And in case of homocystinuria, you will get this, get it at downwards and medially. So please remember the fact, because both homocystinuria and the Marfan syndrome, present you with exactly same symptoms. But what is the difference between them? The first one is the ectopia lentis in case of Marfan syndrome is upward and outward, and in case of homocystinuria, it is downward and medially. Second thing, there is no intellectual disability found in the Marfan syndrome. They will describe you a patient who is very tall, very long arms, and he has a very good grade in his class, and that's how that is a case of Marfan syndrome. But if they will tell you a patient who is very weak, scores very low grades, and the similar symptoms like Marfan, then this must be a case of your homocystinuria. And they will ask you what is the most common cause of death, then it is the aortic dissection. You will study that later in the cardiology section. And you should know that cystic medial necrosis is a normal aging process, but it is speeded up in case of Marfan syndrome. Your arteries become very weak, very easy to rupture because of that. Okay, so any doubt in Marfan syndrome? Anyone? No. Okay, then we will move to the laboratory techniques, and the first one is a polymerase chain reaction. It is a very easy process to multiply the amplify the DNA, multiple like thousands and millions of copies of DNA could be made very easily using this polymerase chain reaction. It includes the 3 steps. And the very important thing that you should know, you do not need to know the whole sequence of the, of the strand of the DNA, but you should know about the sequence of these flank regions. These regions, as you can see here, these are the flank region, and you must know the sequence of this flank region to make this DNA primer. So step 1, we're gonna ask you about this, about the flank regions, what you should know, about the sequence of the PCR in the PCR, and you will know, you will answer about the flank regions. Okay, in order to make the primer, you should know the flank regions of the DNA strand that is targeted. Okay, so the 3 steps: the denaturation, the annealing, and the elongation. The denaturation will takes place at a very high temperature. It will separate out the 2 strands of the DNA, and then comes the annealing. You need to cool down a little bit up to 55 degrees centigrade in order to re-anneal the strands of the DNA. But you do not want that the same strands got re-anneal again, like, right? Okay, so what you will do, you will add a short DNA primer at the flank regions, and at low temperature it will attach to the DNA. That is its complementary sequence. So that's how this works at low temperature. And you should know about the DNA polymerase that works here, which is a Taq polymerase, DNA polymerase. And in the 12th classes, you know that is from the bacteria Thermus aquaticus. In the NCERT book, it is written Thermus aquaticus, but you do not need to know here. Just know the Taq polymerase, which is a DNA polymerase used here. And why we are using this DNA polymerase? Because it can withstand a very high temperature being used in the PCR. As you can see, the elongation requires a temperature of 72 degrees centigrade, and no DNA polymerase can, withstand this much of temperature. So we uses this Taq polymerase because it can withstand very high temperature. Okay, so we put up all our DNTPs inside the PCR, and the Taq polymerase will use these DNTPs to convert them into the DNMPs, and finally will causes the elongation step. And that's how you can make millions of copies of the DNA within very short period of time. The exponential amplification occurs. At one point, there are 2 strands, then become 4 strands, then 4 become 8, 8 becomes 16, 16 become 32, 32 becomes 64, and so on. Okay, so the only thing that you should need to know about this PCR is about these flank regions. You should know the sequence of this flank region in order to make the DNA primers. Okay. Then comes your reverse transcriptase PCR. It is same as a PCR, but instead of the DNA, we use the mRNA here. And using a reverse transcriptase enzyme that I already told you in the telomerase section, a reverse transcriptase uses this mRNA to make the DNA. And the DNA with the same steps of this PCR can be amplified again. Okay. But there's a difference between this PCR and the RT-PCR. That is, in the PCR, you will get these extrons as well as introns. Like I told you, in the gene, there are extrons as well as the introns that will present in this PCR. But because mRNA contains only the extrons, there is no introns. So after the RT-PCR, you will get only the extrons, a real gene, okay, without any interrupting sequences. A real gene you will get after this RT-PCR. Okay, and the DNA is known as a cDNA. Okay, so where you can use this RT-PCR, well, to identify the viruses, the RNA viruses especially, you will use that RNA of the virus, convert it into a DNA. Using the PCR, you will amplify it, and you can know that the virus is present inside our body. Similarly, the CML, BCR ABL gene fusion in the CML, you will get a mRNA after the product, right? And the mRNA could be detected easily. It could be extracted, could be converted into the DNA, and with the help of RT-PCR, you can amplify it. And that's how you can use that the patient is having a CML. The definitive diagnosis could be easily made using the RT-PCR. Okay, so you guys understand this PCR and RT-PCR? Should I move forward now? Or any doubt?
I have a question on BCR-ABL gene. You said that, that the gene for this BCR-ABL will be transcribed into messenger RNA, and then this messenger RNA will be amplified for the detection of CML.
Yes.
Okay.
We will detect the mRNA, then we will convert it into the DNA, the complementary DNA, cDNA, right? And with the help of PCR, we will amplify it. And that's how we know that this DNA exists inside this patient's body. That means the patient is having CML for sure. Definitely diagnosis could be made like that, okay?
Okay.
Then comes the CRISPR-Cas9 technology. A very important thing, I got a question from CRISPR-Cas, and it is very revolutionary technique also. What is CRISPR? CRISPR stands for CRISPR stands for this clustered regularly interspaced short palindromic repeats. You do not need to know this whole name, just know the CRISPR-Cas9 technology. CRISPR is basically a RNA sequence which is complementary to the targeted sequence. Okay, this is a CRISPR, and Cas9 is basically a endonuclease. So CRISPR is the complementary sequence of the targeted sequence that the DNA we want to target, and Cas9 is the endonuclease. So what we do, we will load the CRISPR on the Cas9, and then we will introduce it inside the body. And after identifying its sequence, the Cas9 will do what? It will cleave off the DNA. It will make a single-stranded or either a double-stranded cut in the DNA. It will break the DNA. Okay, after identify that the targeted sequence with the help of this CRISPR. Then what? Yeah.
little bad food hai.
Any problem?
आराम करें.
Any doubt, anyone? Okay, so the CRISPR-Cas9 technology. The CRISPR is basically the RNA sequence of the complementary targeted DNA, and the Cas9 is the endonuclease. Now, after the Cas9 cuts the DNA, it could be done by two techniques. First is the non-homologous rejoining, or second is the homologous rejoining. Like I told you already in the DNA repair chapter, there are two types of rejoining occur: the non-homologous rejoining and the homologous rejoining. Non-homologous rejoining do not require any reference sequence, but the homologous rejoining requires a reference sequence, a chromosome, right? So if the non-homologous rejoining happened, then what will happen? What will happen is basically it will adds up more base pairs, or it will remove the base pair, right? And what ultimately happens is the gene will knocked out, the gene will destroyed, because you are just changing the sequence of the gene now. So after the non-homologous rejoining, what you will left with is a knockout of the gene. And if the homologous rejoining occurs, then what you will do, you will give them a reference sequence, which is our sequence that we're going to give the DNA, okay? And using this reference sequence, it will make the base pairs in the DNA. So what happens now is we have inserted our gene here, as you can see. This is a donor DNA, and after the homologous rejoining, the same sequence will be added here. Okay.
I'm really sorry to interrupt, but can you please repeat this again?
Okay.
This
Again,
Cas9 CRISPR.
The CRISPR-Cas9 technology is basically a revolutionary technique that scientists have developed to alter the gene. Okay, alter the gene. The CRISPR is basically a RNA which is complementary to the targeted DNA sequence. Okay, and the Cas9 is an endonuclease. As you can see here, the Cas9 is an endonuclease, and CRISPR is a RNA which is complementary to the targeted DNA sequence. Now, what happens is this CRISPR will identify the DNA, and after the identification, this endonuclease Cas9 will make single cut or the double cut inside the DNA. Okay, either a single stranded cut or the double stranded cut. Your DNA is cleaved off, as you can see here, divided into two separate segments. Now, these segments can be joined by two types of repair, either the non-homologous repair or the homologous repair. Remember, I told you already in the DNA repair chapter, the two types of repair, the non-homologous and the homologous repair, right? If the non-homologous repair occur, then there is no reference sequence, right? So what the enzymes will do, they will either make more base pairs, or they will remove the base pairs from the DNA. So what will happen is basically you are knocking out the gene, because you are changing the sequence of the gene completely. You're adding the base pairs or removing the base pairs, you're knocking out the gene, right? The gene is no longer function. Okay, you understand this thing?
The non-homologous part that you just shared, that that gene is being changed, the gene sequence is being changed because by non-homologous joining, new DNAs are
Yeah.
being added or or removed.
Okay.
But
Yeah.
I did not understand the other part of it.
Okay. I will explain it again. See, I told you in the non-homologous rejoining that the DNA sequence is not there. Any reference sequence is not there. So what the enzymes will do, they will just randomly insert the base pairs after the, in this segment here. They will randomly insert the base pairs, right? They do not have any sequence, any specific sequence of the gene. So if you're randomly inserting the base pairs, then your gene will no longer be functionable, right? Because there is no sequence remaining in the gene, right? So you're knocking out the gene, you're destroying the gene.
I got it. I got this part.
Yeah. Yeah, good.
Okay.
Now, in the homologous rejoining, you're giving the enzymes a reference sequence, the donor DNA. And using this reference sequence, they will make the sequence here in this part. And because this reference sequence is actually the gene that we want to insert, so after this homologous rejoining, what we left with? We will left with the gene that we want to insert here, right? So this is the homologous rejoining, and we will use this to insert our gene of interest. And the non-homologous rejoining, we use to knock out the gene of interest. I understand that?
Yes.
Yes, got
Okay,
it.
good.
That means if we add the donor DNA, then we will have a new gene that of our interest. But if we do
Yeah,
not add a donor DNA, then this DNA will automatically join itself by non-homologous joining,
yeah,
and the
exactly.
gene will be knocked out. Okay.
Yeah, exactly. And then we will move forward for the blotting procedures. There are four type of blotting procedures: the southern blot, the northern blot, the western blot, and the southwestern blot. Southern blot we use for identification of the DNA, any mutation, any change in the DNA sequence. Northern blot we use for any change in the sequence of mRNA. Western blot we use for the proteins, and southwestern blot we use for those proteins which binds to the DNA, like transcription factors. I told you the enhancer sequence, right? Okay, so southern blot technique basically used to identify the DNA. Now, let us suppose there is a DNA which is a size of 50 kb, okay, and the DNA has a sequence of, known as the palindromic sequence. Now, due to a mutation, let us suppose this sequence is lost, and the 50 kb DNA now become 100 kb size. Now, what we will do, we will, extract out the DNA from the cell. We will destroy it, make it, cutting it into the strands. We will then separate it on the gel electrophoresis technique, separate in the gel, and transport it into a nitrocellulose membrane, which is a radioactive membrane. What we will do now, we will expose this single-stranded DNA to the radio-labeled DNA probes that are complementary to the DNA of interest. Okay, they will bind to that DNA, and we can easily see this radioactivity here. Let us suppose this. Look at this diagram here. These are the parents which are a carrier for some kind of disease. Okay, they have this AA and AA, as you can see here, big and small a. Now, this is the real DNA, the original DNA, which is normal. Okay, it is of larger size. But let us suppose due to a mutation, a palindromic sequence is created inside the DNA. And now this 50 kb DNA becomes a 25 kb size. So what happens is that because we know that there are two copies of same gene is present, right? So the one DNA will move just 50 kb here, and the 25 kb DNA will move more forward here. And similarly with this case, one DNA is here and one DNA is here. Now, let us suppose their children is, having this disease completely, because it is autosomal recessive for this disease, as you can see, homogeneous for this disease. Now, what happens? It has only 25 kb size of DNA, no 50 kb size DNA, right? So their DNA will move here, okay, at a 25 kb size. And that's how you know, because there is no 50 kb size DNA available. This means the patient must be having the disorder. Okay, similarly, this recessive case here, as you can see, this carrier, and this is a normal, as you can see. This is having no symptom. It has only 50 kb size DNA too. You understand this procedure, the sudden blotting? It is dependent upon the creation or the destruction of these palindromic sequences, where you can cut your DNA using the endonucleases. If the palindromic sequence created, then you will get a shorter segment. If the sequence is destroyed, you will get a very larger segment of the DNA, which you can easily separate and identify. Any doubt? Any doubt, guys? No. Okay. So where does you use the Sabin blotting procedure? So any mutation that involves the DNA, you can detect these mutations here, okay, using the Southern blotting. Now comes the Northern blotting, which is basically used to identify the mRNA to check whether the gene is expressed in the mRNA or not, whether the size of mRNA is normal or not, whether the mutation involving the mRNA or not. So basically using the Northern blotting, you can easily detect the mRNA levels. If you know that there is a mutation inside your gene which is not detectable using the Southern blotting, so what you will do, you will wait for the transcription and you will check the mRNA levels, whether the levels are normal or not. If the levels are decreased, that means this must be a promoter sequence defect. The expression of the gene. So whenever you need to detect the mRNA, you will use this Northern blotting technique, like I told you already, right?
Can
This,
you please just repeat? When will we come to know that there is this problem at the promoter site?
if there is a, every gene has a sequence known as a promoter, right? And it is due to this promoter that a gene is expressible, right? If there's a mutation occurs inside the promoter sequence, the gene itself is normal. It is absolutely normal, but due to the promoter sequence defect, their expression will decrease, right? It is like a switch. If the switch is lost, then you cannot just express your gene. But your gene is absolutely normal totally. It just low expression happening.
Then this will be detected by Northern blot, because Southern blot can only detect the gene of the DNA, like the gene itself, not the promoter.
Yeah,
so
exactly.
Because the RNA will not be expressed because of defect in promoter, so we will get low levels of
RNA will expressed, but very low level, at very low levels than what it should be. Okay, and that's how you detect the mRNA levels using the Northern blotting technique. In both the Southern blotting and the Northern blotting, you uses a single stranded RNA or a single stranded DNA, which is radio labeled. Okay, so that you can easily identify the strands in both of these procedures. You should know we will use a radio labeled single stranded piece of DNA that is complementary to the targeted DNA. Okay. And then comes your Western blotting, which is used to identify the protein. So whenever you want to know whether a protein is being made inside the cell or not, whenever you want to know, any defect at the protein level, at the translation level, whenever you want to know the defect at the transcription level, you will use a Northern blotting. And whenever you want to know the defect at the translation level, like the stop codons, like I told you already about the stop codons, if there's a change in the stop codons happens, you can only detect it using the Western blotting, because your mRNA will remains always same, right? So the Western blotting is used to identify the proteins, the protein of interest. What we will use here, we will use the antibodies to detect the proteins, and these antibodies are radio labeled. Okay. For example, your sickle cell anemia. I told you that there is a defect in the amino acid 6th amino acid, the glutamic acid will become the valine, and that can be easily detected using this Western blotting technique, because the change is happening at the translation level, not the transcription level, right? So you will use a Western blot to detect it. Okay, so use the antibody. Now comes the Southwestern blot, which is a very important technique that we use to identify those proteins which bound to the DNA. Like I told you already in the previous lecture about this enhancer sequences, where the transcription factors binds and enhance the transcription of the gene, right? So for the example like C-Jun, C-Fos, and other transcription factors, if we want to identify them, then we use the Southwestern blot. In this technique, what we use are double stranded DNA, because we know that these proteins bind to a double-stranded DNA, right? So we use here not the single-stranded but the double-stranded DNA probes. Okay, you should know this example: the C-Jun, the C-Phos, leucine zipper motifs, and the transcription factors. If we want to identify them, we will use the southwestern blotting. So whenever there is a case of cancer, the proto-oncogenes, if you need to know about this proto-oncogenes, then you should use the technique southwestern blotting, because ultimately all of these oncogenes will use the transcription factors to initiate the transcription and making the cell very cancerous. Remember I told you that these factors can do anything with the cell. You know, they can make a mature cell into a stem cell and a stem cell into a mature cell, right? So it is very important technique. You should remember it. So I hope there is no doubt in this blotting procedures. For the practical implementation, you should use a UWorld. They have very nice questions of them, so you can have a better understanding about these techniques. Okay, or the question will frame they will describe you a scenario of some mutation, some kind of a mutation, and then they will ask you what procedure could be used to detect this kind of mutation, whether it is a southern blotting, northern blotting, western blotting, or southwestern blotting. Okay, they can ask you about this example of C-Jun, C-Phos. These are very important examples you should remember. Okay, so should I move forward now? Any yes or no would be appreciated.
Regarding
Yes, you
several,
can move forward. Sure.
regarding this
Regarding
previous one where the transcription factors were being identified.
yeah.
Yeah. You said that the double-stranded DNA probe is used as a
Yeah.
gene sequence to which the transcription factors can bind. That's how it
Yeah.
happens.
We do not
And
use single-stranded DNA because we know that transcription factors bind to a double-stranded DNA, not a single-stranded DNA, right?
yeah.
So we have to use a double-stranded DNA instead of a single-stranded DNA like southern western blotting or northern this blotting, right?
Yeah.
We use double-stranded DNA here, not a single-stranded DNA. You should remember it, in fact.
And we usually use the genes that are oncogenes because they are very much expressed.
Yeah,
That's what
exactly. Oncogenes actually code for these factors, right? These transcription factors. Yeah. And that's why if you need to identify such type of genes, their transcription factors, you will use this southwestern blotting procedure.
Can
And
you
then comes your
explain
flow.
the zipper motif thing?
These are basically, you have some receptors inside your cell which are carried by these motifs. Actually, you will study that zinc finger motif later in the endocrine chapter, I guess. You will study that later. Zipper motifs. They carries the receptor inside them. UWorld has a very nice diagram of this motifs. Actually, I will show you later. Okay. Then comes the flow cytometry, which is a procedure that used to identify the CD markers. Whenever you see a lab report of a patient in real life also, so the technique is used to identify the CD counts, your CD8 lymphocytes, CD4 lymphocytes. They will list a technique, and they name it a flow cytometry. You can see that also, the flow cytometry. They will written that there. Okay. So what is a flow cytometry? It is a technique that is used to detect CD markers. Whenever you want to know how much CD cells are there inside your cell, what type of CD cells are inside your cells, you will use this technique, this flow cytometry. You do not need to know about this whole technique, but the implementation of this technique. As a diagram here shown here, let me zoom. Now this diagram, as you can see here, this line, before this line, this is 0. Okay. And below this line, this is 0. So these dots represent the density of the CD cells. Okay. So the cell which is positive for both CD8 and CD3, it will shown here. Okay. And the cell which is negative for CD3 and positive for CD8 will be present here. The cell which are negative for both CD3 and CD8 will be present here. And those cells which are negative for CD8 but positive for CD3 will be present here. Okay. As you can see, the data plotted in the histogram, the cells in the left quadrants are negative for both CD8 as well as the CD3. The cells in the right lower quadrant are positive for CD8 but negative for CD3. But you should know that every cell inside your body which is CD8 positive is also CD3 positive. So that's why this area is blank. Okay, and then comes your cell in the upper left quadrant, just here. These are positive for CD3 but negative for CD8. As I told you, below, before this line everything is 0, and below this line everything is 0 for CD8, and before this line everything is 0 for CD3. Okay, and finally the cells which are in the right upper quadrant are positive for both CD3 as well as the CD8. So why the cell is positive for CD3 but negative for CD8 here in this case? Anyone tell me?
The cell has just stirred the subcapsular space of the thymus.
No.
And yet not.
They must be the CD4 cells actually, but they have not plotted this histogram for the CD4 cells. That's why these cells are positive for CD3 but negative for CD8. They must be the CD4 cells. Okay. And every every CD4 cell is also positive for CD3. Likewise, CD8 cell is positive for CD3, right? You guys understand this thing? Flow cytometry. They will give you this histogram and they will ask you what kind of cells are there. Okay,
Yes,
so you should know where is 0, where is 1. You should know that. Okay.
Yes.
Then the DNA microarray. DNA microarray is basically a phenomena. Not a phenomena. A technique where in which you can detect multiple number of genes at the same time, whether the gene is expressing in certain environment or not. Multiple genes, how many genes are expressible at a certain environment. So the technique you use is the microarrays. Microarrays, it is very hard to express, to explain. So I will show you a video regarding this microarray, a very nice video. Okay. So let me turn on my screen sharing. Just wait for a moment, guys. I will share you my screen. Just watch this short video on the DNA microarray. You will understand this procedure better by this way. Okay. You guys can see my screen, right?
Yes.
Okay, let's play this video.
Okay.
Please watch this. Is it audible?
No.
No.
Just a second. How can I make this video audible, guys?
Sir, share this video in the group and later in the if we can see.
Okay.
It is time.
Yeah. Okay, yeah.
Yes, that would be better.
Just a second. Now you guys are able to hear it. Okay, just wait. Is it audible now?
This animation will demonstrate how
Yes, sir.
DNA
Yes,
microarray
yes, we can
experiments
hear.
are performed.
DNA
Please watch this
microarrays,
short video.
sometimes called
The
DNA
clarification
chips,
of this DNA
reveal the
microarray.
expression of thousands of genes on a solid surface, such as a microscope slide. In this example, we'll use yeast as a model system to illustrate one use of microarrays. One common use of microarrays is to determine which genes are activated and which are repressed when two populations of cells are compared. Every gene is measured simultaneously. As an example, we'll compare what happens to yeast genes when cells are grown in aerobic versus anaerobic conditions. The cells grow and adjust which genes need to be activated or repressed in order to survive. Now it is time to isolate the mRNA from both populations of cells. The cells are spun in a centrifuge. Now that the cells have gathered in pellets, we remove the liquid, but not the cells. Next, it is time to extract the mRNA from the cells. When we add the extraction buffer, the mRNA is released into the solution. Next, we remove the RNA and place it in a fresh tube. Now, let's make the cDNA from the mRNA. Here we see 3 of the many mRNA molecules from each tube of cells. Each mRNA is converted into red or green-colored cDNA. When the colored cDNA is made, the mRNA degrades. Then we combine the red and green cDNA, mixing both colors into a single tube. At last, it's time to look at the DNA microarray. In our experiment, a microarray or DNA chip contains about 6,000 spots. Each spot is a different yeast coding sequence from a different gene. Let's choose 3 spots at random to follow in detail. Each spot is made of DNA that can base pair with its complementary cDNA. Here are partial sequences from each of the 3 spots we are observing. Now, let's incubate the mixed cDNA with the DNA chip. For the sake of our example, we'll zoom in and show that some of the labeled cDNA have bound to the DNA in the spots and formed base pairings. Here we see green and red cDNA bound to this spot. Only red cDNA is bound to this spot. And only green cDNA bound to this other spot. In a real experiment, you would not see any of this detail. You would only see the original microarray. Now we must wash off the unbound cDNA to see what is bound to the microarray. Let's detect the bound cDNA so it can be visualized. We begin by placing the microscope slide containing the microarray inside a scanner. We'll examine the next phase of the process, keeping our focus on the 3 spots we've been following. First, a green laser scans the microarray. The resulting image is stored on a computer for later analysis. Now it's time for the red laser. This image is also stored on a computer for later analysis. Now, we move to the analysis phase. After we eject and safely store the microscope slide, we retrieve the red and green images from the computer and create a merged visualization. In the merged image, we see an aerobic gene labeled in green, an anaerobic gene labeled in red, and a gene labeled in yellow that was expressed in both aerobic and anaerobic conditions. This is one example of how DNA microarrays are used. In an actual experiment, quantitative analysis would be conducted on all 6,000 genes.
So you guys understand it now, the procedure of DNA microarray?
Yes.
Okay. So let's get back to our photographs. Okay, so you guys understand the DNA microarray technique now? Any doubt in it? I will share the link of the video again so you can watch it later on again, okay? And now let's talk about the ELISA, which is enzyme linked immunosorbent assay. Okay, this is a technique used to identify either the antigen or the antibodies. Okay, the thing you want to identify, you will extract it from your blood or anywhere inside your body. And what you will do, let's suppose you want to identify a certain antibody inside a human. So what you will do, you will extract a sample of the blood, and you will take a glass or a dish, and in that glass or dish you will uh, attach some antibodies there. Okay, and finally when you put your sample in it, so the antibodies which are which are for this antigen, they will bind to this linked antigen to the glass. Okay, and finally you wash off them, and finally what you will do, you will add some anti-human antibodies which are labeled with an enzyme that is later on used to detect the presence of the antibodies in it. What this antibody does is, yeah, any doubt, anyone? I will show you if second. Just allow it to annotate, right? I will show you with a diagram also. So this is basically your glass, which contain all these antigens. Okay, bounded antigen with this glass. You will put up your sample, and in the sample you will have these antibodies against these antigens, right? So what this antibody is, they will bind to this antigen, right? Similarly, this antibody will bind to it. This antibody will bind to it, right? And now what you will do, you will wash off the sample, and these antibodies will remain bounded to the antigen. Then what you will do, you will add up further the anti-human antibodies that are anti to these antibodies. So if the antibodies are present in this sample, then what will happen is that these anti-human antibodies will bind to these antibodies, right? And these anti-human antibodies will carry an enzyme over them. And when we put another solution over this solution, that enzyme will react with this liquid and it will change the color of the liquid. And after this color is changed, we know that this antibody is in this patient or not, right? As you can see, this is a immunological test. You should detect the presence of either the specific antigen or an antibody in a patient's blood sample. The detection involves use of of an antibody linked to an enzyme, and this is the antibody that is linked to that enzyme. Okay, the substrate, which is a liquid we add, will react with the enzyme, and it produce a detectable signal. Okay, and you should know that it is very highly sensitive and very highly specific, but it is less specific than the Western blot. Western blot is more specific than the ELISA. Okay, any doubt in ELISA? Any doubt?
No.
Okay, then we will move forward. And then comes the karyotyping. Karyotyping is basically a visual representation of all your chromosomes inside your body. Whenever a change in number of chromosomes occurs, you will use this karyotyping to detect it. Okay, and you guys also know the formation of chromosomes occurs in the M phase, right? The condensation of DNA forming of the chromosomes occur in M phase. So for this technique, what you will do, you will let the cell divide. And in the M phase, you will arrest the cell. And after arresting, you will extract out the chromosomes from the cell. And the drug that you use to arrest the cell in the M phase is basically your colchicine. And you should also know that the chromosomes are most easily and very clearly visible in the metaphase. Because as you know, all the chromosomes in the metaphase are aligned along the metaphasic plate, along, according to their numbers. And so they're very easy to identify in the metaphase. So in the metaphase, what you will do, you will add the colchicine inside the cell. And remember, I told you that colchicine is actually a microtubule drug, which damages your microtubule. And I already told you that these, the spindle fibers and the, centromere is actually a microtubule itself. Anything which is moving inside your cell, anything which is moving inside your body is due to these microtubules, these, rail tracks inside your body, right? So we know that in that telophase, these chromosomes will move away from each other, right? Following these microtubules. So what this colchicine do, it will destroy these microtubules and arrest the cell at the metaphase. And in the metaphase, we will extract these chromosomes and we will, number them accordingly. And that's how, if there is a change in the number of chromosome occur. As you can see here, there's a trisomy of chromosome number 5. There's a tetrasomy of number 8, right? As you can see in the arrow. Okay, so any change in the number of chromosome can be detected easily by the karyotyping. And the drug that you use is the collegen, which is the same drug that you use for the gout. Remember, guys?
Yes.
Any doubt in it? I think it's easy.
No. Yeah, this one is easier. Yeah.
Then the fluorescent in situ hybridization, it is the same technique as here, but we will do addition one step. What we will do, we will use a probe, okay, to identify the chromosome so that any translocation, if happens like BCR-ABL fusion, this could be easily detected with the help of this fluorescent in situ hybridization. For example, as you can see, there's a number of, there's a number 17 chromosome, and this is number 19 chromosome. And some part of this chromosome are translocated here. And you can easily detect this part, right? Using this fluorescent in situ hybridization. Any micro deletion happens inside your chromosome, you can easily detect it using the fluorescent in situ hybridization. And you should know that it can detect even a very small deletion. Step 1 gonna ask you this, they will give you an option. It can detect only a large deletion, it can detect a very small deletion, it cannot detect any deletion and something like that. And there you will choose this option. It can detect even a very small deletion. And that's why it is used for microdeletion, even especially for the DiGeorge syndrome. Remember 22q11 deletion, microdeletion in the DiGeorge syndrome, it can be easily detected using this fluorescent in situ hybridization technique. Okay. Secondly, translocation, as I told you, this 17 translocated here at 19, it can be easily detected using this fluorescent in situ hybridization technique. The duplication, if there's extra copy of chromosome, as you can see here in this number 5 and number 9, 8 or 9, 8, the tetrasomy is here and the trisomy is here. You can easily detect it using the fluorescent in situ hybridization. The same technique like karyotyping, but a one step ahead, which is using the fluorescent DNA or the RNA probes. Okay, I think it's also easy. Any doubt? No? Okay, then the molecular cloning. Remember I told you about this RT-PCR technique where you identify mRNA, then convert it to DNA, and then you can know whether this DNA is present or not. And I also told you that the DNA we made after the complementary, this reverse transcription, reverse transcription, this DNA do not contain any introns in it, just the exons, the real gene that we need, right? So what we do now, we'll take up a sample of the human DNA, a gene. That gene contains both the introns as well as the extrons, right? We will let the gene to express itself, the transcription, and when the mRNA forms, it contains only the exons, no introns, right? We will extract out this mRNA, we will use the reverse transcriptase technique, we will convert it into the cDNA. The cDNA lacks any introns, right? It's the real gene that we need. And finally, we will insert the cDNA into this plasmid of the bacteria. And the plasmid can be easily taken up by the bacteria inside it. The bacteria will multiply and then bacteria will, express this gene, making the products. And these products can be easily extracted like insulin and any other drug that we want to, form. This could be easily done using the molecular cloning. You guys understand this procedure?
Yes.
We are done on this page. Now we will move forward, okay?
Okay.
And then we will study the gene expression. I told you about this knockout thing, which is removing a gene from the DNA, right? Knocking in is actually inserting a gene. For knocking out, you need the non-homologous rejoining. For knocking in, you need the homologous rejoining, right? And it is all done on the targeted sequence of the DNA, right? And the one of the technique that I told you is the CRISPR-Cas9 technique. The other technique is a Krylok's technique, which is used for causing the deletion of the gene generally. You don't need to know how this technique works. You should just know that it is used to knock out some genes, okay? Some deletion of some genes which are, causing the embryonic death. I've never faced any question on this Krylok's mechanism, but you should just know that it is used for the deletion of the genes, similar to this, that Cas9 technique, the CRISPR-Cas9 technique. And then an important here, that is your RNA interference. Remember, guys, I told you when an mRNA exits the nucleus and enters the cytoplasm, it will meet up with the P bodies. Now, the P bodies are basically regulatory bodies which can store the mRNA for the later use. They can even destroy the mRNA, right? Using the microRNA technique, miRNA techniques, RNA interference. Now, what happens is naturally your body makes a double-stranded RNA, not a single-stranded RNA. Normally, your body makes a double-stranded RNA, which is which used to shut down the single-stranded mRNA of the interest. Okay, you understand what I'm saying? When an mRNA forms inside your body, there is another mRNA, which is miRNA, double-stranded RNA forms inside your body, which is actually the complementary to that targeted mRNA, and it will shut down that mRNA before the translocation process. So it is a technique which we hacked, and we make a similar RNA, which is a small interfering RNA. It is also a double-stranded RNA, which we made in the lab. We will introduce it inside the cell, and using this small interference RNA technique, we will, destroy the mRNA. We will stop the gene from expressing itself. So what is the proper technique? It's when the miRNA formed inside the nucleus, it will exit the nucleus, enters the cytoplasm. Inside the cytoplasm, it will meet with a protein known as a dicer. The dicer will, cleave this miRNA into short segments. Okay, then it will meet another protein known as I'm sorry, I forgot the name. This RISC complex. After, as you can see, I've written down the whole process here. Just a second. Yeah. So it is a mechanism by which a short double-stranded DNA sequence induced a post-transcriptional gene silencing. It targets the mRNA and destroy it. It inhibits the translocation, not the transcription, but the translocation, which is happening inside the cytoplasm. There are two types of RNAs, and both of them are the double-stranded RNAs, the siRNA and the miRNA. The miRNA is naturally coded by the human body. Okay, it is naturally formed inside our bodies. Okay, and the siRNA is what we made inside the labs. Okay, after being transcribed into the miRNA, it undergoes a processing in the nucleus and then exit out of the nucleus, enters the cytoplasm. Inside the cytoplasm, they meet with a protein known as the dicer. Dicer will cleave them into a shorter segments of RNA helix. Then they will, then the RNA strands will be separated. It will become a single-stranded RNA. That single-stranded RNA will loaded on a complex known as the RISC, the RNA-induced silencing complex. And this complex carries this RNA, which is complementary to the targeted mRNA, right? So if a exact match happens, then it will degrade the mRNA, and if a partial match happens, then it will cause the repression of the translation. Okay, the two things happen. One is a perfect match, and one is a partial match. If a perfect match happens, then the mRNA degradation will happen, and if a partial match happens, then it will lead to the translation repression. I hope you guys understand this technique, this RNA interference. Any doubt in it? Any doubt, guys?
Will you explain
Yes or
that
no?
dicer part once more?
Okay,
Yes,
dicer is
please.
basically a protein. When the miRNA leaves the cytoplasm, leaves the nucleus and enters the cytoplasm, it is very big, right? It is very big. Now, when it meets up with the dicer, it will cleave off into the shorter segments, which are the segments of, which are the segments that are complementary to the, mRNA of the interest. That the sequence that you want to target, the mRNA that we want to target, these shorter segment will carry the complementary sequence of it, right? And that is done with the help of dicer, the smaller segmentation, like a proteasome. Remember, it will destroy the proteins. The dicer will destroy the miRNAs. Not a destruction, but making them shorter. Okay. And then when they become a very shorter segments, they will their strand will be separated. They will become a single-stranded miRNAs, and finally these miRNAs will be loaded on the RISC complex, which is RNA-induced silencing complex. Then they will look for their target sequence of the mRNA. If a perfect match happens, it will destroy the mRNA. If a partial match happens, then it will hold the translocation for some time. Okay. I hope you guys understand this. Any doubt, or you want me to remember? Any doubt, guys? You want me to repeat it again?
No. I think that's okay.
Okay.
You can move forward.
Yes,
Okay.
it's okay.
Now tell me, if the miRNA is formed against a tumor suppressor gene, then what will happen? It will lead to the carcinogenesis, right? So this RNA interference is linked to the carcinogenesis also. As you can see here. Yeah, abnormal expression of the miRNA contributes to certain malignancies by silencing the mRNA of a tumor suppressor gene. And we use the same technique to knock out that the genes for the tumor, right? We use this technique for the tumor suppression. As you can see here, I've written it. The siRNA technique is used for the tumor suppression to knock out the mRNA of the proto-oncogenes. And then we will move for the biochemistry, the genetic terms. I think we should take a short break here, right?
Yes.
Yes, please.
Okay, we will pause the meeting here and we will meet up at 6:05.
Yeah.
Okay with you guys?
Yeah. That's okay. It's
Okay.
fine.
Yeah, average.
And secondly, your voice got
Yeah.
cut in the mid. Can you please your voice got disconnected in the mid? Can you please repeat just the concept of incomplete penetrance again?
Okay, so the
The
incomplete penetrance I was talking about, whenever you have a genotype, this doesn't always means that you get the phenotype. If you have a mutation, that doesn't always mean that you will get the disease. Okay, and this is what the incomplete penetrance means. Penetrance means, the power of penetrance, like that. Okay, so you should know that every autosomal dominant disease, whenever you have this, whenever you heard this term incomplete penetrance, you will always think about the autosomal dominant disease, because autosomal recessive one do not show the incomplete penetrance. It is only for the autosomal dominants. Okay, whenever you have a genotype, this doesn't always means you will get the phenotype always, right? And the percentage or the risk of expressing a phenotype when you have a genotype, this can be calculated by the formula, which is the percentage of the penetrance of the disease of the genotype and the probability of inheriting the genotype. So by calculating it, you can know the risk of expressing a certain mutation. You should know, like whenever you meet a patient with a BRCA1 gene mutation, you just not always do the breast surgeries for the patient, right? Why? Because having a BRCA1 gene mutation doesn't always mean that you will get the ovarian cancer or the breast cancer. And that is the reason why in these mutations, we do not do the breast surgeries always, unless there's a specific risk associated with that. Okay. And then you should know the 3 diseases which shows the incomplete penetrance 100%. Sorry, the 100% penetrance. These are the neurofibromatosis, the Huntington disease, and the achondroplasia. These 3 diseases show 100% penetrance. Yeah.
In autosomal dominant diseases, the penetrance would be high, you mean to say?
No, I'm not saying penetrance is high. It depends whether it is high or low. I'm saying that these 3 diseases have high 100% penetrance, but there are some diseases like BRCA1 gene mutation. They do not have very high penetrance, and that's why having a BRCA1 gene mutation doesn't always means you will get the breast cancer or the ovarian cancer.
What about the autosomal dominant diseases you were saying?
Yeah, I'm talking about autosomal dominant only. BRCA1 is autosomal dominant actually. If you look upon that, it is dominant, not the recessive. Similarly, neurofibromatosis, it is autosomal dominant. Huntington, autosomal dominant. Achondroplasia, autosomal dominant. So this concept is only for the autosomal dominant.
Okay. Thank you.
Then comes a pleiotropy, which is basically when a single gene controls multiple phenotypic effects. For example, the phenylketonuria, you will get the intellectual disability. You will get the the body odor. You will get this fair skin. So when one gene controls multiple phenotypic effect, unrelated phenotypic effects, I must say, then this phenomenon is termed as the pleiotropy. Okay, the example they have shown is the phenylketonuria, which shows the light skin, intellectual disability, musty body odor. Similar to the Marfan syndrome, you will get multiple symptoms. So that so this is a pleiotropy. And there is one more thing I want to talk about, but I'm not seeing it here. You will hear about that later, not in here. Okay, in the reproduction chapter, you will know another term. Okay, so yeah, the pleiotropy. Okay, now we comes at the anticipation, which is remember the disease I told you about, these trinucleotide expansion repeat diseases, the Huntington disease, the Friedreich's ataxia, the Fragile X syndrome, the myotonic dystrophy, where a sequence, a codon repeat itself again and again, like CAG, multiple number of times, 100, 200 times, And in these diseases, what happens actually is with the increased generations, like from grandfather to father, father to child, the number of expansion repeats increases, and hence the disease severity. And therefore, with the increase in the generations, the disease become more severe, and the disease started expressing themselves at very early age. Okay, so increase in the severity and early onset of the disease is known as the anticipation. So whenever you hear about a disease which is a trinucleotide expansion repeat disease, either the Huntington, the Fragile X syndrome, the Friedreich's ataxia, the myotonic dystrophy, they will ask you why the symptoms are keep on increasing, why the severity of this disease keep on increasing, why the and the onset of disease is keep on decreasing. So you will choose the option with the anticipation. Okay, this is due to the anticipation. Okay, then comes the loss of heterozygosity, which is mainly apply at the cancers. So whenever there is a cancer which runs in the family, so the patient might be having a autosomal the patient might be having a I'm sorry about that, I forgot something here. Yeah, so whenever a certain cancer runs in a family, and let us suppose by birth the patient will get a one mutation in one of the two alleles for this cancer, like the patient is heterozygous for a certain cancer. Now once the mutation occur in another allele also, the patient become homozygous right now. Okay, and once the patient become homozygous, he will develop the cancer. And this is what the Knudsen 2 hit hypothesis says. In order to get a cancer, both the both of the tumor suppressor gene must be knocked out, and this loss of heterozygosity is only applied at the tumor suppressor genes. Okay, if you knocked out one gene, then you will still have one gene which is doing a normal function, protecting you from the cancer. Once you get another hit and you will knock out that another gene of this tumor suppressor protein, so you will become the homozygous. You will lose the heterozygosity, and now you will develop the cancer. For example, the retinoblastoma, the Lynch syndrome, the Lee-Fromini syndrome. These are the example of loss of heterozygosity. Okay, so if a patient inherited or developed a mutation in a tumor suppressor gene, the complementary allele must be deleted before the cancer develops. And this is a Knudsen 2 hit hypothesis. It is not true for oncogene. It is only true for the tumor suppressor genes. And then comes a polygenic inheritance, which is basically when a certain phenotype is dependent upon both the genetic factor as well as the environmental factors. For example, the diabetes, the glaucoma, the hypertension, the spina bifida, the cleft lip and palate defect, the balding, male pattern balding. These are all depend upon the family history and as well as the environmental factors. So multiple factors controlling this one phenotype. Any doubt? On previous page?
No.
Okay. So then after that, dominant negative. Yeah.
I have a question.
Yeah.
In in pleiotropy, multiple phenotypes were being controlled by one gene, right?
Yeah, exactly.
Now
multiple
in
unrelated phenotype.
And now in polygenic inheritance, we have a disease, one disease being controlled by multiple factors, be it genetic, be it environmental.
Yeah, exactly. Like diabetes, like balding, like hypertension. You always, hear the patient saying he's having a family history of hypertension. But there's, that not always means that he will develop the hypertension, right? Because it depends upon both the environmental factors as well as the genetic factors. What he eat, how much exercise he do, it all controls the disease.
How did you get a question on that? You were asked to describe the.
They will describe you. They will show you a disease, a symptom of the disease, for example, cleft lip and palate, and they will ask you. They will write a statement from the doctor. What your doctor should be saying to the patient. That it is not always controlled by the genes. Your family history is not that much important, or your family history is important, as well as the environmental factors are important. So that type of statement they will write as an option, and they will ask you which statement is correct regarding this disease. For example, the obesity. There's a question on the ethics, I think, on the obesity, and the patient asks whether my family history plays an important role in this disease. And the option goes to, yeah, your family history plays an important role, but also the environmental factors are controlling this disease too. So that's how you know which diseases are controlled by both environmental factors as well as genetic factors. The list I already told you this: hypertension, the balding, and other diseases.
Okay, thank you.
Then comes the dominant negative mutations, which is whenever there is one mutation occurs and the patient become heterozygote. So what happens is that the mutated gene will affect the function of normal gene. For example, when a p53 mutation occur in one gene, then the abnormal gene will block, or basically disturbs the function of this non-mutated p53 gene. Okay, so this is a dominant and negative mutation. Okay, when a heterozygote produce a normal produce a non-functional altered protein that also prevents a normal gene from functioning. Example, p53 is given. Then comes the linkage disequilibrium. Now, let us suppose there is a allele A and a, yeah.
Can you please repeat that dominant negative mutation, please?
Let us suppose you have two genes p53 and one of the genes become mutated. Now the patient become heterozygote. Okay. Now what happens is that this altered gene, this abnormal gene will also prevent the functioning of this normal gene. Okay, it will make a protein which prevents the normal gene from functioning properly. This is the dominant negative mutation. When a single mutated p53 tumor suppressor gene results in a protein that is able to bind DNA and blocks the non-mutated p53 from binding to the promoter. When this mutated gene binds to the receptor, then the non-mutated gene cannot bind to the receptor, right? Non-mutated protein. And that's how this mutated p53 affects the function of normal p53. This is a dominant negative mutation.
Okay.
Then
Can
comes
you
a
give
linkage.
example of it? Like Marfan syndrome is one that you've written. There's another example.
No, I just know about these two only.
Okay.
I mean, I never encountered a question on this phenomena, so not much examples are given there. But you should know about this p53 for sure. You can encounter a question on that. Then comes a linkage disequilibrium. Let us suppose there are two alleles, the allele A and the allele B. And let us suppose the frequency of allele A is X and the frequency of allele is Y. Now, if the two alleles are free to come together and free to move away from each other, then the frequency of A and B to come together will be XY, right? But the scientists have found that the A and B found in much more frequency together than apart. That the frequency of AB being together will be greater than X and Y. And why this happens? Because A and B are linked together. So whenever cell division occurs, whenever separation of chromosome occur, these two alleles will always go together, and that's why the frequency of occurring of A and B together will be higher than what it actually should be. And this is a linkage disequilibrium. If it is equilibrium, then this frequency AB will be equals to XY. But there is a linkage disequilibrium, then the frequency of AB either the high or lower than what is expected. So a tendency of certain alleles at two linked loci to occur together more or less often than expected by chance is basically a linkage disequilibrium. And you should know that it is not measured in the families, it is measured in a population. And the frequency is always varied. Yeah.
The two alleles to be passed on together is high because they are too much close to each other on the chromosome.
Yeah, exactly. They're too much close on the chromosome and they're linked together. That's why they are occurring more frequently than what they should be if they're free to go, right?
Okay.
Then comes the mosaicism. You should know that mosaicism is a mitotic error, no, not a meiotic error, and the mosaicism cannot be inherited. Okay, what happens is when a cell lineage is kind of mutated during the developmental period, when the zygote forms, it divides into 2 cells, then the 4 cells and the 6 cells, the stem cells, right? And let us suppose one of the stem cells get mutated, so it will create a different lineage of cells inside the same body. Now the patient have 2 type of cells, one with the normal lineage and one with the mutated lineage. So the presence of these 2 distinct cell lines within the same body known as a mosaicism. Okay, so the mosaicism could be of 2 types, the somatic mosaicism or the gonadal mosaicism. Somatic mosaicism occur anywhere inside your body except your gonads. When in your blood, let us suppose there are 2 lineage of lymphocytes are present. One is a X lineage and one is a Y lineage of lymphocytes inside your blood. So this is basically due to the somatic mosaicism, right? And this will affect the patient for sure, right? But in the gonadal mosaicism, it's just your germline, it's just your sperms or the ova that are having 2 different lineages, not your body. So the patient in this case will be normal, absolutely normal, but their child, their fetus, their offspring.
Can someone please mute their mic?
Yes, please mute your mic. Yeah. Mute your mic.
Suhaila is the one who is my song.
I think my eyes is mute. Okay, now the gonadal mosaicism. So in this case, the patient do not develop any symptom because their somatic cells are absolutely normal, but their gonadal cells, their sperm or their ovaries are having 2 different lineages, one with the mutated and one are the normal. So their offsprings will develop the disease, but not the patient itself. Okay, this term as a gonadal mosaicism. And like I said, mosaicism cannot be inherited. Okay, so the patient, the offsprings of the patient will be, abnormal completely, not the mosaicism. Okay, I hope you guys understand what I'm saying. Any doubt in that? The somatic mosaicism and the gonadal mosaicism, it is very important topic.
Can you
Can
give
you
the
please repeat
example of
the last point? Repeat the
Yeah.
last point only.
Okay,
The
I have
genetic.
For the
told you that
last point.
mosaicism, I have told you that mosaicism cannot be inherited. Yani ki, from parents to offspring, 2 different cell lineages cannot be inherited from the parents. In the gonadal mosaicism, even when the offspring inherit the mutation, it will affect the complete body of the offspring. Offspring won't get the mosaicism, but the complete mutation, right? But the patient here, in this case, the father will be absolutely normal, and their multiple child will develop the disease, and that's how you know that this must be the case of a gonadal mosaicism. I've written down the example here. When the parents are all normal and they do not have any mutation or a disease, let us suppose there are 2 parents which comes to you for a counseling of their offsprings. Now you take up their blood sample, you analyze the lymphocytes, you check up their DNA, and it's absolutely normal. No mutations are there. But then what happens is their multiple child, 1 or 2 or 3 child develops the same mutation in them. For example, take up the case of this achondroplasia. Parents are absolutely normal, they are tall, they are healthy, but the multiple offspring have this achondroplasia in them. So that's how you know that this must be the case of mosaicism, because like random mutation cannot be happen in 3 offsprings at the same time, right? So that's how you know this must be a case of mosaicism, and the mutations must be inherited from the father or the mother. So whenever you encounter a patient whose multiple offsprings having the same disease, but the parents are all normal, you will suspect the gonadal or the germline mosaicism. Although the somatic mutations can also happen in the offsprings, but if multiple offsprings having the same mutation, then there are highly chances that this must be a case of gonadal mosaicism. Are you okay with the statement, guys?
Yes, we are okay.
Yes.
I have a question. If the offsprings will have the somatic mosaicism, they will not be able to transfer to their own offspring, right? There needs to be a mutation in the
They will
germline.
transfer the mutation, but not the mosaicism.
Yeah, and there's no. If the somatic cells will have the mutation, they will transfer that.
No. If the somatic cells
No.
have the mosaicism, then it will not be inheritable because only the gonadal cells can go to the offspring, right?
And that's why the parents are not having the disease, but rather
exactly.
the mutations.
Because their somatic cells are absolutely normal. They are having the mosaicism in the gonads only, the sperms or the ova that will inherit in the offspring. So the offsprings will get the mutations, but not the parents in case of gonadal mosaicism. But in case of somatic mosaicism, the parents will be abnormal. They will have the symptom, but their offsprings would be absolutely normal. Okay.
Okay, understood, sir.
Then an example of this mosaicism is the McCune-Albright syndrome. If the question is time ask you if the McCune-Albright syndrome is inheritable, you will say no, because it is an example of mosaicism and it is not inheritable. You should know it is a GS protein activating mutation. It activates the GS protein. You will study about these proteins later in the pharmacology section. Okay. So what are the symptoms you will get in the McCune-Albright syndrome? This is the unilateral cafeolate spots. As you can see here, this kind of a rash, this darkening rash like a coffee. This is a cafeolate spot and it is always unilateral. As you can see, this side is absolutely normal. This side is affected. And their edges are very raggedy. Their edges are not perfect. As you can see, very irregular edges. So cafeolate spots with the ragged edges. Then you will something known as a polyostic fibrous dysplasia, which is the fibrous dysplasia of the bone. Your bone will replace with the fibrous tissue and the collagen. So how you can expect a question on that? They will give you an x-ray where you are showing some where you will see some spots within the hips of the patient, like hollow spots, because the bone will be very bright, the bright looking, but these fibrous areas will be very darker appear on the bone. So that's how you know that this must be a case of polyostic fibrous dysplasia, which is a fibrous replacement of the bone inside the bone. Okay. And the patient must have at least one endocrinopathy, and most commonly the precocious puberty happen in this phase. So whenever you got a patient who is having a history of precocious puberty, and whenever a female has a precocious puberty, and she will always be a very short stature, or the male also, they would be short stature because early closure of metaphysic plate happens in this scenario in the precocious puberty. So the patient must be having a precocious puberty. She must be having a bone pain or easy fractures. Their X-ray will be very abnormal. They will have this atypical rash on the unilateral surface of their body, and that's how you know this must be a case of McCune-Albright syndrome. They can ask you about the mutation. They can ask you whether it is inheritable or not. Okay. So the bone will appear as a osteolytic lesions in the X-ray. I hope you guys know about the osteolytic lesions. Should I show you the X-ray?
The osteolytic lesions are the ones with cavities in the bone.
Yeah, exactly. The same cavities you will see in this fibrous dysplasia. But the fact is the bone is replaced down with the fibrous tissues. So the cavities will appear on the X-ray. The same thing.
So you have the X-ray.
Right.
Can you show that?
Yeah, I can search it on the internet also if you want. I will show you just a second. Okay, you can look at here. These are the osteolytic lesions. I mean, not the fibrous dysplasia, but the fibrous dysplasia will be appear like that only, the darker appearing lesions inside the bone. That's how you know the bone is not here. And along with these symptoms that I've told you, that rash and the precocious puberty, that's how you know that this must be the case of McCune-Albright syndrome. You guys have a very nice question over it. You can see that. they they have shown a X-ray of the hip where you can easily spot the osteolytic lesions in the X-ray, and that's how this must be a case of McCune-Albright syndrome. Am I clear, you guys?
Yes.
Yeah.
Yes. Yeah.
Okay, so very important, McCune-Albright syndrome. It is not inheritable because it is an example of mosaicism. Okay, then these two terms, the aneuploidy and the polyploidy. Aneuploidy means the presence of one extra chromosome only, like trisomy, tetrasomy, right? Or disomy. And polyploidy is presence of complete extra set of chromosomes, which means from 46 to 69. Trisomy of every chromosome is known as a polyploidy. Aneuploidy is just presence of one extra chromosome. And when does this mosaicism occur? Shortly after the fertilization of the egg, so that one complete lineage of cell can be created new. Okay. Then comes the locus heterogeneity and allelic heterogeneity. I think I explained you very clearly already, right? Locus heterogeneity occurs when different mutations occur at different loci or different genes, and they will all produce a same phenotypic effect. For example, the retinitis pigmentosa, the osteogenesis imperfecta, the Ehlers-Danlos syndrome, I told you, multiple genes mutations occur, but they will all produce the same phenotypic effects. And this is your locus heterogeneity. Similarly, the allelic heterogeneity, when multiple different types of mutations occur in the same gene, right? But they will show the same effect. Different mutations occurring in the same gene, but they will show the the same effects. For example, the beta thalassemia, it can have a promoter sequence. Yeah.
Hi, how are you? I left you yesterday. I was exhausted.
Please mute your mic.
Okay, I will repeat it again. The allelic heterogeneity
You're gonna
occurs.
continue, or just because you didn't do the explanation yesterday?
Okay, so I will continue from here. Don't worry, you can cover it. Okay, so the allelic heterogeneity occurs when different.
How about that one from yesterday about the
Yeah, I've told them the example of beta thalassemia.
Yeah.
Right, three type of mutations can happen in the beta thalassemia. One is the promoter sequence defect. One is the splice site mutation, and one other mutation. But all of these mutations will have the same phenotypic effect, the beta thalassemia, right? So different mutations, but same phenotypic effect. This must be the case of allelic heterogeneity. Okay, similarly, the CFTR mutations is also an example of allelic heterogeneity. Multiple type of mutations occur in the CFTR gene, but they will all have this cystic fibrosis only. So these two examples you can remember, and locus heterogeneity, you can remember the osteogenesis imperfecta, the Ehlers-Danlos syndrome, the retinitis pigmentosa, and the albinism.
In locus heterogeneity, we have different loci. In allelic
Different loci mean different genes.
Different genes. So in retinitis pigmentosa, there are different genes involved.
Yeah, exactly. Multiple type of genes, multiple type of mutations, but all have a same phenotypic effect.
And in allelic heterogeneity, it's just the same gene but different mutations.
Single gene. Exactly. Same gene, different mutations.
Like when you said the promoter site mutation of beta thalassemia, the gene is same. The splice site mutation, the gene is same, but the location within the gene is different.
Yeah, exactly. The type of mutation is different, right?
Okay.
Then comes the heteroplasmy, which is basically takes you to the mitochondria. The presence of any mutation in the mutated mitochondria inside a cell is termed as the heteroplasmy. Okay, when you have this normal mitochondria as well as a mutated mitochondria, you will say it is a heteroplasmic case. And the severity of disease depends upon how much mutated mitochondria you are inheriting from the female. As you guys already know that males cannot transfer their mitochondria to the fetus. Only the females can transfer their mitochondria. So this heteroplasmy is totally dependent upon the females. Whenever a female is affected, all of their offsprings are affected. When the male is affected, no offspring would be affected, right? And the severity of the disease is totally dependent upon how much mutated mitochondria you are inheriting, right? You should know about this heteroplasmic term. Whenever you encounter a disease, which is a mitochondrial related disease, for example, Leber's hereditary optic neuropathy, Mela syndrome, or the Rett fiber syndrome. These three disorders, we will encounter them later here in this. I think in the next page, you will hear that this heteroplasmy. They will ask you a question, what about the severity of the disease? What factors the severity of disease depends? So you will say that it depends upon the number of mutated mitochondria, and the term is known as a heteroplasmy. Okay. And then comes the uniparental disomy, which is basically when single parents transferred both the chromosomes. Okay, when a single parents transfer 2 copies of the chromosomes and no copies from other parent would be present. For example, you have you will hear about 2 disorders, which is Angelman syndrome and the Prader-Willi syndrome, where only the male or the female will transfer their chromosome. So whenever 2 copies of the chromosome came from only one parent, not both the parents, this term as the uniparental disomy. They will try to trick you in the case. They will give you a case where one male is like heterozygous for some autosomal recessive disease, and the female would be absolutely normal, and then somehow you will get the offspring with the disease, right? And that's how you know that there must be a case of uniparental disomy. Both of the abnormal gene must be come from the same male parent. Okay, I hope you guys are understanding me. It's a bit complicated thing, right? They will describe you a scenario where the male patient is having an autosomal recessive disease or the genotype of the autosomal recessive disease, and female will be absolutely normal, but somehow they will get an offspring having the disease. And that's how you know that this small a or the abnormal mutated gene must be transferred. Both the copies of this gene must be transferred from one parent only, the male. That's why the offspring would be having this disease. And the examples of these 2 disorders of this uniparental disomy is basically your Angelman syndrome and the Prader-Willi syndrome. You will study about them in later next page, I guess. Then comes the Hardy-Weinberg population genetics. Very important, guys. They will describe your scenario and
before moving towards the other, can you repeat the heteroplasmy?
Heteroplasmy, I told you, is the presence of both abnormal as well as the normal mitochondria. And the severity of disease depends upon how much mutated mitochondria you are getting inside your cell. You are inheriting from the female, because the mitochondria can only be inherited from the female. Male do not contribute to the inheritance of the mitochondria, right? So if a female is affected, then it will transfer its mutated mitochondria to the to the generations, right? And the severity of this disease is totally depend upon how much mutated mitochondria you're inheriting from the female. As you can see, the first aid writes a line that mutated DNA passed from mothers to all the offsprings, but the severity depends upon how much mutated DNA is being passed on. So this is the heteroplasmy. Severity depends upon the portion of abnormal to the normal mitochondria in the patient cells. Okay, so is that clear now?
Yeah.
Yes.
Okay, so now the Hardy-Weinberg principle, they will give you a scenario where they will give you a certain number of individuals with a disease, certain frequencies of a gene, and they will ask you how much is the chance that a offspring will inherit the disease. Okay, I will show you a calculation, but I don't know how. Okay, so let us suppose there is a family, or there is a population where the frequency of of a disease is basically 0.2. Let us suppose. Let me open this pen thing. Okay, let's suppose there's a population where the individuals having the disease frequency is basically 0.2. Okay, and then they will give you something like a case. So yeah, anyone wants to say something? Anyone? No. Okay, I was hearing some voices, so I thought, okay, so let's suppose they will give you a case where the frequency of certain disease is basically 0.2, and they will tell you that this must be a case of autosomal recessive disease. So what you will do is 0.2, because it is an autosomal recessive. So you create a box, and this 0.2 correspond to the frequency of this small a and small a here, right? And this will denote as this Q square. Q square is basically the frequency of inheriting the small a and small a, and the patients, all the patients with this small a and small a must be having the disease, right? So the 0.2 is actually your Q square. And by default, because these diseases are very rare, as you can see, there's only 0.2% chance of inheriting the disease. Okay, so by default P square will be equals to 1. You will always write that P square will be equals to 1. Okay, now you will see that formula, this P square plus 2PQ plus Q square equals to 1. They will ask you the carrier frequency first. What is the chances of being a carrier? So they've already given you this key in this Q square. This Q square will be equals to 0.2. So you should need to calculate the frequency of Q only, not the Q square, but just the Q. So the Q will be equals to under root of 0.2, right? 0.2. Now, what is the carrier frequency? This 2PQ you are seeing here, this 2PQ is actually the carrier frequency. You need to calculate this. And I've already told you that P square must be 1, so P will be equals to 1. So 2Q will be equals to 0.2 under root. Sorry, this 2Q, you need to calculate this carrier frequency, right? And Q, we already know that this must be equals to 0.2. So the carrier frequency must be equals to 2 under root of Q, which is 0.2. So that's how you know this is a carrier frequency. I hope you guys understand this equation. You just need to know that P plus Q. P is the normal gene, Q is the abnormal gene. This is equals to 1. So the P square will be equals to the having a normal allele, which is A, and Q square is basically having the Q square is basically a frequency of having a disease, which is small a and small a. 2PQ is basically the carrier state. So they will give you the frequency of certain disease, and they will ask you what is the carrier frequency. The carrier frequency must be equals to 2PQ. With the help of frequency of the disease, you will first calculate the Q, which is a small a allele, and then you will calculate the 2Q, which is 2 under root of this thing. Okay, so understand the calculation, guys?
Yes.
I can repeat it again because it is very important. You can expect at least one question from this Hardy being the principal.
Okay.
And then they will ask, okay,
So
should I repeat it again?
Can you repeat, please?
Okay, so let me take a pen and a paper. Wait for a second, guys. I will open my camera. My video is visible or not, guys?
Yeah, it's visible.
Can you see the pen and the paper?
No. You can see your
Pen and
face.
paper are not visible.
Now?
Yes.
No. Pen and paper are not visible.
You can.
We
sir,
cannot
you
see.
You can just open the notepad on your computer and then just.
It's actually kind of very difficult to write it on the laptop actually.
Yeah, we can see your. We
You
can
can
see
open my
your
screen.
paper.
Yeah, everyone can see my paper.
Yes.
Yes.
Yeah, we can see. Yeah.
Okay, so let us suppose they will give you an example of a certain family. Okay, and the family is having is from a population where a frequency of certain disease is basically 0. Let us suppose 0.16. Okay, this is a frequency of having a disease in a population. Okay, they will tell you that the female is a carrier of the disease. Okay, the female is a carrier of the disease. So the carrier status of the female is already known, which is small a and small a, right? And they will ask you if a donor from this population is taken, then what are the chances that the offspring will get the disease? You understand my question?
Yes.
They will tell you about a population where having a certain disease is basically whose voices is. They will ask, so they will give you a population where the frequency of certain disease is given, and they will tell you that a donor is selected from this population. They will tell you that female is a carrier of the disease, and if they mate, then what is the chances that the offspring will get the disease. So what you will do first, you will use the Hardy-Weinberg principle, which says that the frequency of double, इसको मैं write करता हूँ, this double A will be equals to P. This is basically your 2PQ, and this, sorry, it is P square, and this small a will be equals to this Q square, right? So they have given you the frequency of the disease, which means the Q square, because having a disease means you are having this genotype, right? So what they have given you this Q square already. So Q square will be equals to this 0.16. Okay. So now you need to calculate what are the frequency of having allele small a, or the Q. That will be equals to under root of 0.16 or 0.04, right? Okay. So now you got what are the chances of having this Q in a population. Now you need to calculate what are the chances that this donor is actually a carrier of the disease. So how we can calculate this? This is the carrier status, 2PQ. We already know that P is basically a very large number. This must be a 1, because having a disease is very rare in this population. 0.16 is nothing. So you can just write it as 1 always. You need to write this as 1. So now we know that P square equals to 1, or P is equals to 1. So 2PQ can be reduced to 2Q only, right? And this would be equals to 2 into 0.04, which is 0.08, right? So having a carrier frequency is basically 0.08. These are the chances that this donor is a carrier. And we already know that female is a carrier. The carrier frequency must be equals to 1. So the carrier frequency of female is 1, and the carrier frequency of male or a donor is equals to 0.08. Now, if they have an offspring, let us make a Punnett square. This is a female status. This is your male status. Okay. So this is a chance that their offspring will be abnormal or having the phenotypic character, which is 1/4, right? And what is the frequency of having a male as a carrier? This is equals to 0.08. And what is the chances that female is a carrier? This is equals to 1. Now what you will get, what are the chances of offspring being a diseased, is basically 1/4 multiplied by 0.08, which is the chances of male being a carrier, multiplied by 1, which is chances of female being a carrier. This will be equals to 0.02. So they will ask you this question only. This is the hardest question which could be made on this Hardy-Weinberg principle. I hope you guys understand this calculation, right?
Just please repeat once more.
Okay, so they will describe your population where they have given you a chances of the diseased log. Okay, they will give you that certain disease having a frequency of itna in a population, right? Then they will ask you if a male donor is taken from this population, then what are the chances that offspring of a carrier female and this male donor will be having the disease. So first of all, you need to know if the donor is a carrier or not, right? So you already know that the frequency of being a diseased is 0.16. And when you will get the disease, when you will get the allele small a and small a, which is the Q square actually, or you can write it as A square. Both are same, right? Now you know that the Q square is basically equals to 0.06. And what are the chances of having this A allele? Chances of having this A allele, that is basically Q equals to 0.04 after the under root of 0.16, right? Now you need to know the carrier status, and I've already told you that this 2PQ is being a carrier. The chances of being a carrier is 2PQ. P will always be equals to 1 because, the chances of being normal is almost equals to 100%. So you will always write P equals to Q. So now being a carrier of the disease will be equals to 2Q. So the 2Q will be equals to 2 into 0.04, right? Here, this will be equals to 0.08. So this is the chance that this male donor is a carrier. And what is the chance that this female is a carrier will be equals to 100%. They have given this to you, the 100% chance of being a carrier in the female. So now if you make a Punnett square of these two patients being a carrier, this will be equals to the frequency. This is the chances, 1/4 are the chances for this fetus, to be having the symptoms of the disease, right? And this is 1/4. 1, 2, 3, 4 scenarios are possible, and only one scenario he will having the disease. But what are the chances of male being a carrier? 0.08. What are the chances of female being a carrier? Equals to 1. So you will multiply these carrier status of the father and the mother, and you will get the answer of 0.02. This is the hardest question they can make on this Hardy-Weinberg principle, actually.
Thank you, sir.
Okay. Anyone want me to repeat it again?
No, that's okay. I think we can move forward.
Okay. Okay, so please be careful with this Hardy-Weinberg principle. You can surely expect at least one question from this part. Okay, let's move forward. And yeah, one more thing for the X-linked diseases, for the X-linked diseases, if the male is basically having the disease, then it is not the Q square, but the Q, because male has only one chromosome, right? One X chromosome. So if a male is having the disease, that doesn't mean that he's having a Q square, he's having a Q. He just need one small a, right, for having a disease. So it is not Q square, but just a Q. So if they give you a frequency of disease in a population and they tell you that it is an X-linked disease, so that this disease frequency is equals to Q. And now you can check the carrier status of a female by applying this formula 2PQ, where Q equals to the number of patients having the disease, or the frequency of patients having the disease, right? So be careful about that. You will get some questions in the UWorld, then you will have a better idea of how this thing works, okay?
Okay.
Then the disorders of the imprinting. What is imprinting, guys? I told you in the last lecture, in the very first page, about this methylation, right?
Yeah,
So
the gene copies get silenced.
Yeah, the gene
Because
copy
of the
gets silenced, and that
methylation.
is done by
Because
the
of the
methylation.
methylation.
Where does the
Yeah.
methylation takes place?
It can be
On the
DNA
cytosine.
methylation. Oh yeah.
On the cytosine, right? Yeah. So now this is the practical implementation of this methylation, actually. So what you will see, these two syndromes, the Prader-Willi syndrome. Yeah.
It was cytosine and guanine, or not only cytosine?
Yeah, cytosine, cytosine and adenine for the prokaryotes, and only cytosine for the eukaryotes.
Okay.
Can you repeat what thing, please?
Okay, so methylation of, or the imprinting, will takes place on the cytosine in the eukaryotes, and on the cytosine as well as adenine in the prokaryotes.
Okay, thank you.
That's what I was saying. Okay. Now, the Prader-Willi and the Angelman syndrome both takes place in this chromosome number 15. You should know the chromosome number, very important. Now you should know on this chromosome there is one gene which is silenced in the male, chromosome, and there is one gene which is silenced in the female chromosome by default. Okay. Now let us suppose if two copies of chromosome number 15 came from only one parent, which is male, then both the genes by default will be imprinted in the male. So the offspring will lost both the copies. You guys following what I'm saying? This is a default scenario in our body, that in the chromosome number 15, one copy of, one gene from this chromosome will be silenced in the male, and another gene will be silenced in the female chromosome. By default it is, right? Now these syndromes will happen when both of the genes will be knocked out, and this could happen when either of the chromosome is inherited from one parent only. Okay. You guys understand what I'm saying, or should I repeat myself again?
No, I think it's okay.
Okay, now the Prader-Willi syndrome will occur when both the copies came from female, because there are some genes which are imprinted in the female chromosome, and if both the copies came from the female, then both of the gene will be knocked out, right? And that will lead to the Prader-Willi syndrome. Or sometimes what happened, because female copy is already knocked out by default, if somehow male copy is already deleted by some kind of a mutation, then you can also get this Prader-Willi syndrome. Okay, so maternally derived genes are silenced already, and if the parental allele is deleted, then you will get this Prader-Willi syndrome. What are the symptoms of this syndrome? There is this hyperphagia thing. Okay, these patients are always hungry, and that's why they will get very obese. They will have very high BMI. They will get this intellectual disability, the hypogonadism, and the hypotonia. And one more thing, they are very short stature. So whenever they describe you a patient having all these symptoms, you will think about the Prader-Willi syndrome. They will ask you the cause of it. There must be a parental deletion, or either that both of the copies came from the maternal origin. That is uniparental disomy I've told you about in the previous page. Okay, you should know the chromosome number, which is chromosome number 15. Okay, then comes the Angelman syndrome. In this syndrome, what happened? Either the female copy is deleted, because by default, male copy of this gene is being deleted already. So if the female copy is also deleted, or both the copies came from the male, then you will get this Angelman syndrome. In this case, the patient will be having seizures, and they will always, keep on laughing at inappropriate scenarios. So whenever you get a patient who is having seizures and he's very happy baby, you will think about this Angelman syndrome, and they will ask you the cause of it. So the cause is basically the deletion in the maternal copy of the chromosome number 15, or either both the copies came from the parent. Sorry, from the male parent. Okay, you should know the chromosome number. That is most important thing. Any doubt in this disorder, guys?
So they can ask you either which one was mute or where both coming from. It
Yeah,
could be
exactly.
like two different questions.
In Prader-Willi, the deleted copy would be of male parent, or either both copies are from the female parent. In the Angelman syndrome, either deleted copy is of female parent, or both copies came from the male parent.
Okay.
You should know the number of chromosome, which is chromosome number 15. Okay. As I told you, as I shown you here, this is a chromosome number 15, and these darker are the imprinted genes. By default, these gene will get imprinted. So you will get one active gene in the male copy and one active gene in the female copy. If somehow one of these will knocked out, then you will get this Prader-Willi or the Angelman syndrome. Now comes the mode of inheritance. First one will be the autosomal dominant. If in case of autosomal dominant, one parent have the disease, then there are 50% chances of being inherited by this parent. 50% of the offsprings will be affected. And you should know, in case of autosomal dominant scenarios, no parental line, no generation would be left unaffected. Every generation would be affected. If you're getting a generation which is not affected, then this must be either the case of autosomal recessive scenario, or this must be a incomplete penetrance case. Remember, incomplete penetrance, when you're having a genotype but do not develop the phenotype. So two scenarios could be possible in the autosomal dominant. Okay, you should know just this, the fact that if one of the parent is affected having the dominant gene, then almost 50% of the offsprings will be affected. You should know the example of all the autosomal dominant diseases. We will study them later in the next page, the examples, but just know about it. Okay. Then comes the autosomal recessive condition. If one of the female or the male is a carrier, then there is just 25% chances that their offsprings would be affected by this disease. There are 50% chances that they must be the carrier, and there is only 25% chances that the offspring would be normal. As you can see, out of these three, this one is affected, right? And you should also know that they will show you the skip generations, which means that it is not necessary that all the generations are affected. There will be some generations left without any phenotypic effect, and then suddenly that disease will re-emerge in next generation. And that's how you know that this must be a case of autosomal recessive. Okay. So when two carriers or heterozygous are the pair, and you should also know that for disease being transferred to the next generation, for the offspring should be affected, both the parents must be having a carrier status. Okay. The male and the female both must be carrier in order to transfer the phenotype to the next generation. Otherwise, it would not be possible. Okay, so when two carrier or the heterozygous parents, on average, one of the four children would be affected, homozygous for a certain trait, 50% of the children would be carriers, and 25% of the children would be neither affected nor the carriers. You should know this fact only. Then comes the X-linked recessive. Okay, so for the X-linked recessive, you should know the male parent can only transfer their X chromosome to the female. Okay, no male to male transfer occurs, and female, one copy will be transferred to the male and one copy will be transferred to the female. So if male is affected, male father is affected, then no male offspring would be affected. And if a female is affected, then there is 50% chance of being a transfer the disease to the offsprings. The 50% of the female must be carriers, as you can see, 50% are carriers, and 50% of the male offsprings are affected. There is no male to male transmission occurs. So if they are showing you a pedigree where if male is affected, then no offspring would be affected, or no male would be affected, then this must be a case of your X-linked recessive disease. You should need to identify by looking at the pedigree what is the X-linked, where it is autosomal recessive, whether it is autosomal dominant. By just looking at the pedigree, you should need to identify the thing. Okay, that's how a question would be presented. Then comes the X-linked dominant scenario. Yeah.
So if a mother has homozygous disease, homozygous genes, what are the chances in the son having the disease?
If the female is having homozygosity, right?
Yeah.
You can draw a Punnett square again here. So let us, it's just a second. So if the female is homozygous and male is normal, you are saying, right?
Yes.
This is our homozygous female, the capital X and the capital X. This is your normal male, the capital X and a Y. Okay, so you will get a capital X and the capital X. This is small x. Okay, the male is normal, right? So this is small x, not a capital X. I'm sorry. The male is normal. Small x is normal, and big X is meaning you are having a defective gene. Okay, so small x. So capital X, small x means a carrier female. Again, a big X, capital X, and a small x, which means again a carrier female. So there are 100% chance that female must be carrier, as you can see. Male, there are 100% chance that male must be affected with the disease. Male must be having a phenotypic trait of the disease. Okay, as you can see, both the males are affected. Right?
Okay.
So that's how you can draw a Punnett square, and you can identify the chances of being a carrier of the disease, or chances of being affected with the disease, chances of being the normal. They will ask you to calculate it. They will give you a family history of certain disease. You need to identify whether the disease is having a carrier status or not.
Excuse me.
Yeah.
Hello. Can you tell me about the incomplete penetrance and the relevant pedigree?
Yeah, I was talking about it. If you get an autosomal dominant scenario, for example, and you are sure that this disease is an autosomal dominant because multiple generations are affected, but somehow you will get a skip generation. Somehow one generation would left unaffected, and then again the next generation would started having the disease, and almost 50% of the offsprings are affected with the disease. That's how this must be a case of autosomal dominant disease with less than 100% penetrance or the incomplete penetrance, right? So you should know in this scenario, you should be able to differentiate between the autosomal recessive and the autosomal dominant with incomplete penetrance, because it will look like same. almost. But in autosomal recessive, you will get only 1 or 2 generations affected. But in autosomal dominant with incomplete penetrance, you will get only 1 or 2 generations left unaffected. Rest all the generations would be affected continuously.
Okay, thank you.
Class charge
So the skip generations are because of the
charge
incomplete penetrance.
I'm hearing multiple voices.
It's me, Cameron. I'm asking.
Someone please mute your mic, please.
Neha, can you please mute your mic? Yeah, I have a question, and that is the skip generations are because of incomplete penetrance, right?
Yeah, exactly. Skip generations. Yeah, exactly. Okay, so now the X-linked dominant, and you should know if mothers are affected with the X-linked dominant, then there are 50% chances of both daughter and the son being affected. As you can see here in this scenario, the mother is affected here, as you can see, and there's 50% chances that both male and female are affected. 50% chances. And if a male father is affected, then there is 100% chance that a female would be affected and 0% chance the male being affected. Because I already told you there is no male to male transmission occurs in the X-linked dominant. And you should know the name of 3 syndromes or diseases who's showing the X-linked dominant rate. And the first one is the fragile X syndrome. We will study that later. The Alport syndrome. Remember the collagen number for Alport syndrome? Anyone?
I4.
Collagen number 4. Yes, exactly. And one of them is a hypophosphatemic rickets. Like you already know that the bone is made up of calcium and phosphate. So if you have a deficiency of vitamin D, then you will lack the calcium and phosphate, right? And what you will get is a rickets. Now, in this case, in this autosomal dominant disorder, hypophosphatemic rickets, you will get a hypophosphatemia in your blood. There is a phosphate wasting occurring at the proximal tubules. And as a result, you will get very low phosphate. Your bone will become very big and the presentation will be same as a rickets. The bone pain, the fractures, the bowing of the legs. Okay. So this is hypophosphatemic rickets, the same as rickets, but it is due to the low phosphate content in your body, and it is an X-linked dominant disease. Then comes a mitochondrial inheritance. Remember what I told you already about it, the heteroplasmy. It applies here, right? As a first aid says, all offsprings of affected females may show signs of the disease. If a female is affected, then every individual or every offspring of the female must be affected. So if you are getting such type of, pedigree, then you should know that this must be a case of mitochondrial inheritance. For example, here in this case, the female is affected, as you can see. And as a result, all of the offsprings of this female are affected. Similarly, the female here is affected, and as a result of all of the offsprings are affected. But as you can see, the male is affected here and the female is normal. So no offsprings are affected. So you can just repeat this term. If female is affected, then all offsprings are affected. If you're seeing this type of pedigree, then this must be a case of your mitochondrial inheritance. Okay. And the severity of the disease depends upon how much amount of mutated mitochondria you're inheriting. Okay. And this terms as the heteroplasmy. And the 3 diseases are possible with this mitochondrial inheritance. The first one is the Melas syndrome, which is mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. The second is basically your myoclonic epilepsy with raked fibers. And the third one is your Leber's hereditary optic neuropathy. You can write them down. First aid has already mentioned only the 2 diseases, which is Melas syndrome and Leber's hereditary optic neuropathy, but there are 3 diseases, 3 different diseases. First one is the Melas syndrome. Second one is the myoclonic epilepsy with raked fibers. And third one is the Leber's hereditary optic neuropathy. Now, as the name suggests, Melas syndrome, it is myopathy, mitochondrial myopathy with encephalopathy, the lactic acidosis, and the stroke-like episodes. So whenever you encounter a patient having multiple episodes of strokes, having lactic acidosis inside the body, or having some CNS disease, anything like any, psychiatric disorder, anything like that, then you should know that this must be a case of Melas syndrome, and they will ask you what type of inheritance pattern they show. So they will show the heteroplasmy. Okay. Secondary. This is the myoclonic epilepsy with raked fibers. You will see the multiple episode of epilepsy in these patients, and the microscopy of this patient will show you the raked red fibers. Why these raked red fibers are showing? Because of the accumulation of diseased mitochondria. You should know the mitochondria give it, the red color. So after the accumulation of diseased mitochondria, you will see the muscle fibers being very red. So they will write this term, this raked red fibers, and that's why you should, this must be a case of mitochondrial inheritance. And they will ask you where these mitochondria are being accumulated. Then this is the subsarcolemma of the muscle fiber. And they will give you a strain for staining these red fibers. And this is a Gomori trichome stain they will use for muscle staining. Gomori trichome staining is used for the muscle staining in case of mitochondrial syndromes. Okay, the third one is a Leber's hereditary optic neuropathy, in which you will get the blindness. Because it is optic neuropathy, it will affect your optic nerve. This causes the death of the optic neurons and will, it will cause the blindness. So we'll describe you a scenario where the patient is having a blindness, and on the, on the fundus view, what you will see is the pale optic disc. Whenever they tell you about something like that, this pale optic disc, there must be a case of optic neuropathy. This is how they describe the optic neuropathy, the pale optic disc. Okay, so whenever you encounter a patient having a bilateral vision loss and a pale optic disc, you should know that there is a, and along with having symptoms like these myopathies, like myopathy, muscle pain, having some sinus diseases, having some raked red fibers, or something about the Gomori trichome staining, or some epilepsy thing. So that's how you know this must be a case of mitochondrial inheritance, and the heteroplasmy should come into your mind. And they can also ask you that what kind of functions must be affected. So you should know that some of the tRNAs and rRNAs are also made up in the mitochondria. Okay, so some of the tRNAs must be lost, their functions might be affected, and now you also know that the oxidative phosphorylation runs in the mitochondria, right? So because of these mutated mitochondria, your oxidative phosphorylation is lost, and that's why you are getting this lactic acidosis inside the body. Okay, and as you know, whenever there is a lactic acidosis happens, the first organ will be affected will be the organ which utilizes more oxygen. And because this organ is not utilizing oxygen now, you will get the symptoms. For example, the brain, it requires so much oxygen at a time, but there's just deficiency of it. So you will get the serious diseases like strokes and some epilepsy and other things. Any doubt in it, guys? It is so much important to know.
Because brain uses a lot of oxygen, that's why you said that the central nervous system will be more affected.
As you can see, I have read it.
Is affected,
is lost
because
mitochondria are mutated.
Yes, and what is the impact of tRNA, rRNA in the manifestation? There is no manifestation.
There might be some, because you're having some, they're having a lot of symptoms, right? And some of them might be due to the tRNA, because of the fact that tRNA is involved in the protein synthesis. You are losing the protein synthesis to some extent, but not much effect, but yeah, some effect. Actually, there was a question in the step 1. They have asked about it in the mitochondrial inheritance. They, they show a scenario where the patient is having these symptoms, and they've asked that why the patient is having these symptoms, or what functions must be affected due to this scenario. And they didn't write an option about this oxidative phosphorylation, but they wrote an option saying the tRNA. So that's why I have mentioned about this tRNA thing, because tRNA is affected in this mitochondrial inheritance thing. Not just the oxidative phosphorylation, but tRNAs are also affected. So you must know this fact.
Question.
Yeah.
There are mitochondrial diseases, will always see it when they are in kids, or can they stay and then manifest
even in
later in life?
the kids and in the adults, it depends upon how much mitochondria you're inheriting, right? I told you the severity of the disease depends upon the mutated mitochondria, right? The heteroplasmy. If it is more heteroplasmy, more mutated mitochondria are there, that then it will present in the kids also. If you're having very few mitochondria, then it is very, it will present in the later life.
Okay.
And they've asked a question about it because, They will describe you a family where the father is having this symptom and the offspring is having this symptom, the grandmother having these symptoms, and they will ask you why such widespread array of the symptoms are there. Why the spectrum of symptoms are there, why the patients are not having similar symptoms. So the answer will goes to the heteroplasmy. Because of the heteroplasmy, the number of mitochondria will be different in different individuals, right? And the symptoms will totally depend upon how much mutated mitochondria you are getting from the female.
Okay, thank you.
Okay. Any doubt in this page, right, guys?
No.
No.
Okay, so now you should know the examples of all these autosomal dominant diseases, and the easy way to learn it is that the fact that all the cancers inside your body are all autosomal dominant. All the BRCA gene mutation, the TP53, the Lee-Fraumeni syndrome, the Lynch syndrome, right? All of the cancers are the autosomal dominant diseases. Okay. Plus the neurological, there's a table in the FA about on this neurofibromatosis, Sturge-Weber syndrome, right? Osler-Weber-Rindau syndrome. There's a table on it in the neurology section. All of the diseases are autosomal dominant. So as you can see, the achondroplasia I told you already, the achondroplasia and this this table of Huntington's disease. Remember this table here? The neurofibromatosis, Huntington disease, achondroplasia, they show 100% penetrance. So you at least know these three examples. And now all these cancers inside your body are autosomal dominant. So you can learn from here, right? See, the neurofibromatosis type 2, the tuberous sclerosis, the von Hippel-Lindau disease, the neurofibromatosis type 1, they are all listed in one table in the neurology section. And they are all the autosomal dominant diseases, right? The Huntington disease, the Lee-Fraumeni syndrome, the Marfan syndrome, okay? The familial adenomatous polyposis, which is actually your GIT section, the cancer, the GI cancer, right? The hereditary hemorrhagic telangiectasias, okay? The Lynch syndrome, all of the cancers are autosomal dominant. That's how you can learn these examples. There is no easy way. I'm sorry, guys. Yeah.
The hereditary hemorrhagic telangiectasia, it's not a cancer, right? I just have to.
Yeah.
Just wanted to.
I'm telling you a way to remember these things. All the cancers are autosomal dominant. First thing you need to know, okay? So whenever they describe you a scenario of a cancer, and if they ask you what type of dominance pattern they show, you should know that this must be a case of autosomal dominant without any doubt. There's a table in the first aid on the neurology section about this neurofibromatosis and other disorders, and all of the disorders listed there are autosomal dominant, except one, which is Sturge-Weber syndrome. It is mosaicism actually. Okay. Similarly, the Huntington disease, the achondroplasia, I told you 100% penetrance. These are the autosomal dominant diseases. Okay. The Osler-Weber-Rindau syndrome is autosomal dominant. The cancer, this familial adenomatous polyposis, it can lead to the cancer. It is autosomal dominant. The autosomal dominant, as the name suggests, in this polycystic kidney disease, it is autosomal dominant, right? So that's how you can easily learn these examples, because you should know these examples. You need to remember these examples. So that is the easy way to learn them. Then comes the autosomal recessive diseases. And there's no easy way. You should learn them just like that. I will skip that part, okay? Because there is totally learning, remembering thing. Rectification.
Yeah,
Okay, now the cystic fibrosis. Okay, so cystic fibrosis is basically a mutation in the CFTR gene, which is located in the chromosome number 7. It is an autosomal recessive disease, as the name suggests, okay? And you should know where the deletion occur. It is phenylalanine 508 position. Phenylalanine 508th position, the mutation occur here. And likewise, I told you that multiple sites of these genes could be affected by the mutation. And that's why it is an example of allelic heterogeneity.
Yes. Yeah.
Okay. there are more than 100 type of variants of cystic fibrosis exist. More than 100 type of mutations have been identified on the cystic fibrosis, and all of them are on the CFTR gene on the chromosome number 7. So this is an example of allelic heterogeneity. Now you should know that CFTR encodes for an ATP gated chlorine channel. Okay, you should know that there are some channels inside your body, which transfer the chlorine out of the cell or inside the cell depending upon the location. Okay, when we talk about the respiratory system, okay, then this chlorine must be exported out of the cell. But when we talk about the GI tract, then this chlorine must be absorbed in the GI tract through these channels. But what happens is, because CFTR is lost now, so the chlorine will remain trapped inside the cell and cannot go outward. And as a result, to maintain the electrical neutrality into the cell, the sodium and water will rush into the cell, okay, making all these mucus and your secretory products very dehydrated. And as a result, you will having all these symptoms in the cystic fibrosis due to these dehydrated, very hard products of the secretions. It will causing the duct blockages, and due to these duct blockages, you are having these symptoms. We will study about them later in these paragraphs. So you must know what kind of mutation is most common type. So the most common type mutation is a misfolded protein, where the protein just retained in the rough endoplasmic reticulum and cannot transport it into the cell membrane, because you know that the chlorine channels are present in the cell membrane, right? So due to this misfolding of the protein, mishandling of the protein will remain in the rough endoplasmic reticulum and cannot go to the cell membrane, and as a result, the channels are lost. The chlorine will be trapped inside the cell, and as a result, intracellular chlorine will leads to the increase in the sodium absorption and as well as the water absorption. And this leads to the dehydration of the mucuses and all secretions inside your body. Okay, like stools, your mucus, your pancreatic secretions, your lung secretions, your liver secretions, your bile secretions, everything will become very thick and very hard, that it will lead to the duct blockage. And all the symptoms you are facing in the cystic fibrosis are due to this fact that it is causing the duct blockage. Okay, now because sodium is being reabsorbed into the cell, it will causing more negative transepithelial potential difference, more negative potential around the cell membrane. Okay, and because every sodium inside your body is being delivered inside the cell, what you will get is a hyponatremia. So the one of the treatment of the cystic fibrosis is giving the patient too much salt, sodium chloride, so that hyponatremia could be treated. Okay, and due to this hyponatremia, one of the common symptoms you will get is very lethargia, lethargy, this nausea, this CNS symptoms like drowsiness, very weakness you will get. Okay, so this is the pathophysiology of this cystic fibrosis. Now, how will you diagnose the disease? Because I already told you that two type of channels are exist depending upon the location. For example, in your lungs and respiratory tract, the chlorine will be secreted out of the cell through these channels. And in your GI tract, the chlorine is absorbed into the cell through these channels. Okay, and likewise your sweat. In your sweat, the chlorine is basically absorbed into the cell before the sweat is take place. Okay, so what you will do, you will do the pilocarpine test. Now you should know the pilocarpine is basically a muscarinic agonist. What it will do, it will inducing the sweating. It will induces the sweating, and as a result, you will check the chlorine content inside the sweat. What you will get, because I told you that chlorine must be absorbed inside the cell while sweating, but because chlorine channels are inhibited in this case, the chlorine will not be get absorbed inside the cell, and as a result, it will lost in the sweat. So you will get a very high concentration of chlorine in the sweat, and that's how you know the patient might be having the cystic fibrosis. Secondarily, because I told you that all of the sodium will get reabsorbed, so you will get the hyponatremia, and water will always follow the sodium. So what you will get a alkalosis and a contraction. Remember, the RAS system will get activated because of low sodium delivery, and what it will cause? It will cause the alkalosis and the hypokalemia also, along with this hyponatremia, because there always gonna be a potassium loss along with the sodium loss. Okay, and that's why the FA says the effects are analogous to a patient taking loop diuretics, because you're activating your RAS system because of low sodium in your body. Your RAS will get activated. It will lead to the
Can you please repeat the link of RAS system here?
Okay, so you know the RAS is totally dependent upon the delivery of sodium to the DCT, right? You know that, okay? Because in your whole body, the sodium is absorbed into the cell. There's already a hyponatremia in your body. So a low sodium delivery will be happen in your DCT also. That will lead to the activation of RAS, right? And whenever the RAS is activated, it will lead to the loss of potassium from the body, right? So that you can get the hypokalemia. You will study that later in the renal section, how you will get the hypokalemia due to the activation of the RAS, and you will also get the alkalosis because of this RAS. Okay, so you can measure the body pH, you can measure the potassium level, you can measure the sodium level and the chlorine level in the body. You will get very low level of these things, and that's how you know this must be a case of cystic fibrosis. You understand it now?
Yes, I did.
Yeah, RAS activation totally depends upon the sodium delivery. Okay. And second, because you know your secretions are all affected, all of your enzymes which are being secreted in in your GI tract will be affected. So one thing or one important test you guys should remember is the immunoreactive trypsinogen test. This test we use for the newborn screening. The question stem might show you a case of a patient with cystic fibrosis. They will not tell this is cystic fibrosis. They will show you some symptoms, and they will tell you that this guy is having a immunoreactive a trypsinogen positive, and that's why this must be a case of cystic fibrosis. Okay, please remember this test, immunoreactive trypsinogen. This is very important. Okay, so now let's come to the complications of this cystic fibrosis, because I told you all of the complications are due to the fact that your secretions are very dry, very thick, very hard. Okay, so because of the blockages in your respiratory tract, you will get some infections, recurrent pulmonary infections in the kids and early childhood. You will get the staphylococcus aureus infections in the respiratory tract, pneumonia, recurrent pneumonia due to the staphylococcus aureus. In the adulthood, you will get the pneumonia due to Pseudomonas aeruginosa, and you will also get the allergic bronchopulmonary aspergillosis, which is basically a hypersensitive reaction against this fungus, this aspergillosis. A hypersensitive reaction against the aspergillus fungus. So you will get the recurrent attacks of something like asthma attacks, recurrent, without having asthma actually. And ultimately what will happen, because of this recurrent inflammatory attacks, your all these airways will become very dilated, and you will get a chronic bronchitis and the bronchiectasis, which is the dilation of the airways, the bronchiectasis. Okay, you can also see the involvement of the sinuses. You will get recurrent sinusitis. You will get a recurrent otitis media because of thick secretions inside your middle ear cavity and thick secretion inside your sinuses. So guys, you can see that all of the respiratory symptoms are similar to that of this primary ciliary dyskinesia. Remember the cartesian syndrome? The symptoms are very similar in both of the disorders, right? So how will you differentiate between the two disorders? So the first thing is in the
pancreatic involvement. By pancreatic involvement.
Yeah. First involvement is the GI symptoms. If you're getting obstruction in the GI passage, or maybe a diarrhea, then you can think of the cystic fibrosis. If they will describe you a patient which is a failure to thrive case, the patient is having very low BMI, he is very having very low height, and a patient with malnutrition, then you must think about the cystic fibrosis, because failure to thrive is not present in case of this the cartesian syndrome. Okay, so the two things, one is basically the GI symptoms, and second is the failure to thrive. These two things will differentiate the cystic fibrosis from the cartesian syndrome. Okay, then because you are losing all your secretions, all your fat, because there is no protein, you need the enzymes to absorb your fat from the GI tract, right? But I already told you that all the enzymes and all the secretions are being very dried and very hard. You cannot get the absorption of fat in your GI tract, and as a result, all these fat soluble vitamins like vitamin A, D, E, and K will be lost from your body. Okay, the pancreatic insufficiency occurs because of these thick secretions in your pancreas. Someone is saying something?
It's noise background somewhere.
Okay. So I told you because of these thick secretions inside your pancreas, you will get water blockage in the pancreatic ducts and you will get a pancreatic insufficiency. And whenever you get a pancreatic insufficiency, no fat enzyme could be delivered to the GI tract, and as a result, you will get the loss of all the fats, and because of loss of fat, you will get a steatorrhea. You will get diarrhea, which is rich in very fat. You will get the malabsorption of the proteins. You will get the malabsorption of the fat, malabsorption of these fat soluble vitamins, because of the blockage of the biliary duct, you will get the biliary cirrhosis. Because of blockage in the liver, you will get the liver disease. And you will get something known as a meconium ileus. Because of this very hard and thick stool, because there is no water in the stool, your stool become very thick and very dry, very hard. So it will lead to the blockage of the GI tract. And this will cause a very, a pain, constipation in the newborn. So either you get a diarrhea or you get a constipation in case of cystic fibrosis. Constipation with lots and lots of pain and bloatings. Okay. And one of the important thing that is seen in the cystic fibrosis is the infertility due to the absence of vas deferens. Remember, in the Cartnager syndrome, you are getting the infertility because of this immobile sperms, right? But in case of cystic fibrosis, you will get infertility due to the absence of vas deferens. But you should know that spermatogenesis will be absolutely normal. It just your sperms cannot be delivered because of this absence of vas deferens, and that's why you are getting infertility. So it could be a scenario of a male patient who is having, who is having a thing of this infertility, and then you will look upon it, and he will give you the history of recurrent, lung infection or failure to thrive. He will have very low BMI. He will get recurrent diarrheas, and that's how you know this must be a case of cystic fibrosis. Okay. And another thing you will get is the nasal polyps and clubbing of the nails. Because of this bronchiectasis, the CO2 amount in your body will start accumulating very high, right? And you will get very altered, blood vessels inside your lungs. And because of this altered blood vessels, you will get this clubbing of the nail. I will describe you why you get this clubbing in the nails in the respiratory section. But you should just know because of this bronchiectasis and bronchitis, you will get the clubbing of the nails also, because of low oxygen in your blood. Okay. I hope you guys understand why the symptoms are occurring in this cystic fibrosis. Any doubt?
Yes.
Yeah. What doubt?
Excuse me.
Because of low oxygen, the clubbing of the nails occurs.
Like actually, whenever there is a change in the blood vessels inside your lung happen, it will always lead to the clubbing of the nails. Okay, it is somewhat related to the megakaryocytes actually. It is related to the megakaryocytes actually.
Okay.
Yeah.
Would you repeat that immunoreactive trypsinogen testing?
Yeah, in the exam scenario, they will describe you a patient having some symptoms of the cystic fibrosis, and they will tell you that the patient is positive or having an increased immunoreactive trypsinogen. And that's how you should know that if they've given you this buzzword, it's like a buzzword, okay? If they have given you, then you should know that this must be a case of cystic fibrosis. It will help you identifying the disease if they will give you. So you should know about this immunoreactive trypsinogen test, which is used for the cystic fibrosis. Most of us will missed it, I myself, during my preparation, I missed it many times. In my fourth pass of the first aid, I came to know that this test exists. So you should know about this test also. You can expect 1 or 2 questions in emboss from this. Okay. So now comes to a treatment of this cystic fibrosis. The first thing is the saline, because you're having very dehydrated secretions from the body. So the first thing you will do is a hypertonic saline. You will increase the tonicity in your extracellular fluid. Okay, that's the first thing. You will give the patient albuterol to dilate your airway so that you can breathe very easily. Okay. The second thing is basically azithromycin, which is used as an anti-inflammatory agent in this case, the cystic fibrosis. And one of the important drug that you use in the cystic fibrosis is ibuprofen. I'm sure most of you guys missed it many times, so you can write it down. Ibuprofen is used for the treatment of cystic fibrosis. It will slow down the progression of the disease. Then because you are losing your pancreatic secretions, pancreatic enzyme, the pancreatic enzyme replacement therapy is also an option. And you should know this pancreatic enzyme replacement therapy must be given orally, not the IV route. Generally, we give whenever we give an enzyme replacement therapy, we'll always give it through the IV route. But here in this case, the pancreatic enzyme therapy will be given oral route. Why? Can anyone tell me? Generally, we give the enzyme. Yeah.
We give generally IV because they will be metabolized in our GI.
Okay. And why in this scenario?
Because we
Why
want them to work in the GI.
Because we want them to work in. Because we want them to work in the GI.
Yes.
Right? That's why. Okay, so now pancreatic enzyme replacement therapy, and there are two drugs, the lumacaftor and the ivacaftor. We also use for the treatment of this disease. You should know the name, lumacaftor and the ivacaftor. Okay, the lumacaftor actually changes the folding of the protein. It will correct some misfolding of the proteins and improve the transport of this CFTR protein to the cell membrane. And the ivacaftor actually helps in opening the chloride channels. Okay. And as you can see, the salt supplementation is also recommended because you are having this hyponatremia thing, right? And you will also get the pancreatitis, a chronic pancreatitis because of the cystic fibrosis. And this chronic pancreatitis leads to the pancreatic insufficiency. You will study that later in the GI section also about this pancreatic insufficiency, the chronic pancreatitis. Because of this blockage of the ducts, your enzymes will get activated inside the ducts also, and this will create some inflammation inside your pancreas. So you should know this fact also. And here in the first aid, they have shown you a diagram of the CFTR channels. As you can see in the sweat ducts, normally the chloride and the sodium get absorbed. But due to this cystic fibrosis, the channels are lost. And as a result, the chlorine and sodium is losing from your body through the sweat. And so that you can measure this content of chlorine and sodium inside your sweat. And if it is increased, it will indicate a cystic fibrosis. In your airways, as you can see, the chlorine must be secreted out of the cells to maintain normal mucus, a hydrated mucus. But due to this loss of channels, the chlorine is trapped inside the cell, and sodium and water will also come inside the cell to maintain the electrical neutrality. And as a result, you will get a dehydrated mucus, which will block your air passages. Okay. So this is about your cystic fibrosis. Any doubt?
You have written something regarding octreotide. What you would like to
where?
say? Octreotide, somatostatin analog. You have written something
on the left of this page.
Yeah, okay. This octreotide. This is a fact actually you need to remember. Octreotide is basically a somatostatin analog, and it is contraindicated in the cystic fibrosis, but because it will aggravate the symptoms. Because you know all these somatostatin analogs will inhibit the pancreatic enzymes. You know, guys, right? So you're already getting the pancreatic insufficiency, and above that, if you give a patient octreotide, it will just aggravate the symptoms more. Okay, so this octreotide is basically contraindicated in this scenario. Okay. Move forward now. This is a difference between the primary ciliary dyskinesia and the cystic fibrosis symptoms. Okay. The extrapulmonary defects are very important because you will get a sinus invertis in the primary ciliary dyskinesia, but a pancreatic insufficiency in case of cystic fibrosis. In both cystic fibrosis and dyskinesia, you will get the infertility, but infertility in dyskinesia is due to the immobile spermatozoa, where in this cystic fibrosis, it is due to the loss of vas deferens. You will get a failure to thrive in case of cystic fibrosis, but no failure to thrive or normal growth in the primary ciliary dyskinesia. Okay. Then some symptoms of this cystic fibrosis, the rhinosinusitis, which typically present you with a facial pain. I already told you in the last class about that. How will you say that patient is having a sinusitis? They will present you with a facial pain and running nose, nasal congestion. And how will they describe you a nasal polyp? They will tell you that, they're having some kind of a growth inside the nose mucosa, a glossy, shiny mass, they will tell you. And that's how you know this must be a case of nasal polyp. Okay, then X-linked recessive and X-linked dominant diseases we will study. Should we take a break now, guys?
Yes, I think we should.
Okay, 10 minutes break.
Yeah.
We'll meet
Okay.
again at 8. Okay.
Okay.